that purpose, we used BLBC mice for the experiment, and we inoculated the infection by CR1 strain that was cholesterol-resistant strain. As you can see, there are 4-fold and 2-fold rise in those mice that were infected by 2 strains, and the CR1 strain that was resistant to colistin. It increased the neutrophils, but not to that much extent. There was a decrease in bacterial colonization of liver, lungs, and spleen caused by CR1 or the colistin-resistant strain. Now, as the treatments for colistin-resistant are very limited, so we were interested to explore the effects of Caesuria lepa, that is a traditionally occurring medicinal herbal plant in Asian region. For that, we prepared the water extract of this herb, and we saw the effects of Caesuria lepa in, again, those mice. These are the different strains that were used to cause the infections in mice. And as we expected, the Caesuria lepa, it showed antimicrobial and decreased inflammation in the neutrophil counts, WBC counts, and those mice that were treated with this herb, they showed decreased bacterial colonization in the liver, lungs, and spleen, again in the mice. As evidence that opaque colonies of AB5075 strain of Acinetobacter, it is more virulent as compared to the translucent. So we again used these 2 strains, and as compared with the previous results, again Caesuria lepa, it produced anti-inflammatory effects and significant decrease in the neutrophils, WBCs, and again bacterial colonization in the respective organs of the mice. The conclusion was that out of 150 samples, 84% were resistant to carbapenem groups, 7.3 showed resistant to colistin, and when we found the whole genome sequencing and multi-locus sequencing, there were novel point mutations in the Acinetobacter bimini 17978. In addition to that, the CR1 strain, it produced virulence, but to a lower extent in the mice model. And last but not the least, the water extract of Caesuria lepa, it showed an anti-inflammatory response and also decreased the bacterial colonization among different organs in the mice. The data is published in Frontiers of Pharmacology on 16th of September. And if you want to know any details, the data is published online. Thank you so much.
Thank you, Dr. Mehra Ehsan. Our next oral presentation is on analysis of fluconazole resistance and mutations in ERG11 gene of Candida albicans, and the presenter is Sadaf Zaka.
Assalamu alaikum. This is Sadaf Zaka from Aga Khan University Hospital. I have been working on antifungal susceptibilities of yeast and mold, and this presentation is one of its components. So today I am presenting analysis of mutation in ERG11 gene of fluconazole resistant Candida albicans. So Candida albicans normally colonize human body, mostly in skin, gastrointestinal tract, vagina, and oral cavity. However, low immunity, long-term use of antibiotics, and HIV, diabetes, or any invasive surgical procedure could cause fungal pathogenesis. If we look into the data from Pakistan, 2006 till 2019, we have not reported any fluconazole resistance. Even in 2020, we have not seen any resistance. But from 2021, this fluconazole resistance is raising its head. Recently, WHO has released its list of 19 fungal pathogens, and interestingly, Candida albicans fall in the critical priority group. not just because of its public burden, but because of its fluconazole resistance. So let's see the mechanism of action of azoles. Candida albicans, being a eukaryote, have a unique cell membrane possessing sterols. Main esterol, ergosterol, it provides stability and flexibility to cell membrane. Lenosterol is a precursor. Okay, so what does azoles do? It binds to the active site of the enzyme and causing depletion of ergosterol, resulting in the breakdown of cell membrane, and eventually organism dies. There are several molecular mechanisms which Candida albicans have developed to encounter drug effect, mainly mutations in ERG11, some efflux pumps like ABC promoters and MFS, and overexpression of ERG11 expression. These are the mutations reported in several papers globally in azole-resistant isolates. So what was the objective for our study is, we have seen resistance in Candida albicans, so we analyzed the ERG11 gene for mutations. We collected samples from routine laboratory sampling and then identified by biochemical identification process and conventional methods. This diffusion susceptibility was done, MICs were determined, and ERG11 gene was amplified, and then Sanger sequencing was done. MEGA11 software were used for sequence analysis. So here are the results. We concluded the data from 2021 and 2022, 3 2024 isolates from 2021, and 2 446 isolates from 2022. And if we see the specimen distribution, non-invasive specimens and invasive specimens. So, in this chart of specimen distribution, less number in 2022 doesn't mean that we obtain less number of albicans in 2022. This is just the data of 9 months. And mostly albicans came from a female, and the age group from which we get most albican is adult. So susceptibility testing was done from a few of the isolates, and out of them, in 2021, 2 came out resistant in fluconazole, and 2022, 4 came resistant to fluconazole, and 3 to Vori. And we did MICs for them, and result is in front of you. Blood, pus, CSF, and HPS harbor these resistance. Out of these 5, we analyzed 3, and other 2 are in process. So upon analysis, we have got E266D and K342R in HPS specimen, and blood specimen harbor T123I and Y132H. So if we look into the reported data, this E266D and K342R is normally not found in resistant isolates, but we have seen in our resistant isolates. So we need to look into the other mechanisms of resistance in it, because the strain which has this mutation is not just resistant to fluconazole, but it showed resistant to all azoles. Yes, and this one is interesting mutation, T123I and Y132H. This double substitution generates a synergistic effect on rate of resistance, and it increased the MIC level to 8 microgram. And it is normally found in resistant isolates. So what we conclude from this study is Candida albicans, fluconazole resistance is emerging. So we need to explore more isolates in MICs and analyze gene so that our healthcare system has much evidence to treat patients effectively. Thank you.
Thank you, Sadaf. Our next free paper is on the frequency and susceptibility profile of Listeria isolation from clinical specimens submitted at a tertiary care hospital of Pakistan from 2016 to 2022 by Dr. Imran Ahmed from Aga Khan University Hospital.
Assalamu alaikum everyone, and thank you so much Dr. Munir Bahasan for introducing me. So what I'm going to talk today, before that I begin, this is not a very in-depth analysis. This is a preliminary analysis of a study that we are currently undertaking. So what I'm going to talk about today is the frequency and susceptibility profile of Listeria monocytogenes isolated from clinical specimens at our laboratory, and the time period is from 2016 to 2022. I don't have any conflicts of interest to disclose. So as we all know that Listeria monocytogenes causes listeriosis. It's a rare but can be life-threatening disease in certain individuals and a subset of population. It's found almost everywhere in the environment. It's a gram-positive bacillus. It has a characteristic tumbling motility that we use to identify it. And it's present at room temperature but not at 37 degrees centigrade. It's a very characteristic feature of this organism. It not only can survive at refrigerated temperatures, but also can multiply and can reach to concentration that can really actually reach to its infectious dose. It is highly salt tolerant. It can tolerate also the high and the low pH, so alkaline and acid pH do not actually kill it, and it's an intercellular pathogen which are motorized by its various virulence factors that it has. It is acquired mainly through ingestion, but other routes also do exist. For example, zoonotic and vertical transmission is documented. Farm animals, soil, vegetation, these are the main reservoirs of this organism. Even today, it has high mortality. 20 to 30% has been reported in the literature, even with adequate antimicrobial therapy. Listeriosis also affects animals. It's commonly known as silage disease and manifests in various entities. So, for example, meningoencephalitis, abortion in animals, perinatal death, septicemia in newborn animals. In humans, listeriosis can be divided into 3 categories broadly. So for non-pregnant patients, pregnant patients, and for in the unborn and the newborn child. So if you look at the non-pregnant patients, it mainly it's very uncommon disease, but it's mainly mainly manifests as gastroenteritis. Its self-limiting incubation period is quite short, and a certain individual compromise may be at high risk of. For the pregnant ladies, you can compare that this subpopulation is at a higher risk, much higher risk of getting this infection. Incubation period, if you compare it with the non-pregnant population, it's quite prolonged. So from 70 to 67 days, even 70 days have been documented in the literature. Diagnosis is difficult because most, if the pregnant lady gets this infection, the only symptom may be just a fever-like illness and flu-like illness. So it's difficult to diagnose in this population. In fetus, if it's a vertical transmission, or it's the baby can acquire while passing through the birth canal. If it's acquired in utero, so miscarriage, stillbirth can result, and in the newborn, bacteremia, septicemia, meningoencephalitis can result, and it may be divided into early and late onset listeriosis depending on when the symptoms develop in the newborn child. So there's plenty of data available on the international from the various countries around the world. So from US, UK, Southeast Asia, China, and there are various reports available from our neighboring country, India as well. When I looked at the local data, although there is availability of listeria prevalence in the foods that are sold in the open market, in the public market, but I couldn't find any data on the human cases of listeria from Pakistan. So as you can see from Quetta, Hyderabad, and from Punjab, there are reports that the food that are sold in the open market have listeria monocytogenes in them, so various proportions. Why did we get to start this study? So we weren't seeing listeria monocytogenes infection in the past. It's been a rare sight, and because we don't have documented literature available from Pakistan, and in the recent years we have seen a rising trend in its isolation from clinical specimens, and that's why we started studying this pathogen. So our objective was to describe the isolation, frequency of isolation, and susceptibility profile of listeria monocytogenes from human specimens that are submitted to our laboratory from 2016 to 2022. So we looked at all the cultures that were submitted during this period, and also we looked at our PCR assay that is done in the and it's commonly known as BioFire, meningitis encephalitis panel, so FAME panel, that was introduced back in June 2017. We also performed the antibiotic susceptibility, and it was done for ampicillin, meropenem, and cotrimoxazole. It was done on Mullihenton agar supplemented with 5% sheep blood, and we did it with, by distribution. So a total of 92 isolates or 92 reports, 92 listeria monocytogenes were reported during this period out of 2 million cultures and BioFire specimens. The BioFire specimen reported 9 listeria cases from CSF, and out of these 9, we were able to grow 8 of them. 60% of these isolates were from female patients, nearly 60%, and you can see the distribution. It's mainly CSF and blood, but we had 2 cases from placenta, from aqueous humor, ascitic fluid, and brain abscess. So total of 92 cases. If you look at the age distribution, you can see 3 bumps that are usually reported in the literature. So neonates, the 19 to 45 years age, I will come to it later, and the elderly. Our 2/3 of neonatal infections were early neonatal sepsis, and if you can see 19 to 45 years, 80% of these patients were female. So quite similar data from what has been reported in the literature. So as you can see, a quite remarkable increase in the isolation rate of this pathogen. From 2016, there were none, coming down to 2022, and up till October 2022, we had reported 36 in these 10 months, in these 10 months. And it was so interesting that we went back and looked at the data, and we went back as far back as 2007, and we found none of the listeria monocytogenes reported during this period. If you look at the geographical distribution, although there were 92 specimens, but 44 specimens were from our hospitalized patients, so they would be qualified. So we looked at the residences of, or where the sample was submitted. So we looked at outpatient data, and 48 specimens qualified as outpatients. So majority came from Karachi, but you can see from Karachi to Gilgit and to Peshawar, and here is the graphical representation that these specimens are coming from all over Pakistan. When talking about the susceptibility profile, we reported 92 during this period. One, as I told you earlier, one did not grow on culture. That was reported on biofire, and ampicillin could not be tested for 2 cases. So here's the susceptibility profile. So nearly 100% susceptibility to ampicillin, meropenem, while for cotrimoxazole it was 82%. So this study shows very clearly that there's a rising trend. It's an emerging pathogen actually in this part of the world. Although the incidences from UK and USA are reported to be stable, but from various countries, including Asian and European countries, there's a or either stable or an increasing trend has been reported. Although increasing trend has been reported, but there's not much debate on why this is occurring. So we do not know if it is climate change, if it's dietary practices, and we know that in Pakistan, ready-to-eat food, deli foods have been introduced, but either there are no standards or we fail to follow those legislations to comply with the quality of a standard that are required for the storage, for the production of these ready-to-eat food items. And our data agrees with what is reported from around the world. So it clearly demonstrates that this pathogen is on an increase, and it's an emerging pathogen from this part of the world, and ampicillin still qualifies to be included in the treatment regimen along with other agents. So we need to increase awareness among microbiologists and clinicians to keep it as a differential in appropriate patient populations. So prevention is what is required, and through improved food safety in the production line, in how the food items are stored, and increasing awareness among clinicians, microbiologists, and the general population. So we plan to extend this study by doing serotyping, molecular sequencing to see the relatedness and establish collaborations with our veterinary sector and determine the mechanisms of antimicrobial resistance in these important pathogens and describe some clinical features. So these are my references, and thank you.
So thank you so much. This is the end of our this session. So I'm very grateful to all the speakers, and I will invite Dr. Afia as a first chair to come for the concluding remarks. Sorry for the question answer session, because you can ask the question answers during the tea break, because the short of the time we are cutting down that part. Dr. Afia, can you come and for the concluding remarks, and then there is a distribution of shields also. Thank you.
Thank you very much, speakers and oral presenters as well as participants, because it's very depressing if we don't find appropriate participants. But what I believe about that 100% of you are microbiologists, so I'm very happy today. In the morning I was not, but now it's very good crowd over here, and thank you. Yes. So there were, in the beginning, there were 3 invited talks. All 3, they presented their case very intelligently, and Dr. Irfan Mirza, he talked about the pan-resistant organism and the problem we are facing from the laboratory side. Dr. Faisal, in his usual style, he discussed the simplest approach, how can we approach patients, because many of them are there. And Dr. Altaf Ahmed, he talked about infection control. And 3 of them, I think 3 of them, they agreed that it should be integrated approach, and it, otherwise we can't fight them. Then the 3 oral presenter, I'm very happy. Actually, being a panelist, it is very difficult. One was PhD student with her thesis. The second one, Sadaf, is a very young research associate, and Dr. Imran, the experienced assistant professor. So it's very difficult to give the mark, but I'm very happy that all three, they presented their papers very well. And thank you very much for the questions. Questions were very targeted, and I believe they were very pertinent. So thank you very much, everybody. And so we will do the distribution of prize very quickly. Otherwise, अगला session.
Yeah, madam, please आप आ जाइए ताकि आप दे दें.
Shobha?
Dr. Shobha, आप भी आ
Shobha
जाइए,
ji,
आप भी आ
Shobha ji,
जाइए.
Shobha ji.
Okay, so our first presenter today is Professor Dr. Irfan Ali. Huh? किनको? Madam, आप. अच्छा, madam, बैठिए. हाँ. Dr. Iram, Dr. Afi, आप लोग आ जाएं, बिल्कुल, madam. So our first talk speaker is Professor Dr. Irfan Ali Mirza. Professor Dr. Faisal Mahmood. Professor Dr. Altaf, I think, चलें उनको बाद में दे देते हैं, सही है. So हम लोगों को भी और आप लोगों को, okay, पहले मैं.
Dr. Faisal Hani for moderating this session.
डॉक्टर शोभा इसके बाद आप आएंगी. And finally, Dr. Afia as a chair. Tea is in the Zevar Hall. Kindly proceed for the tea and come back after half an hour, please. Thank you. 11:30 by 11:30.
मैंने दे दिया डॉक्टर को.
बता दिया.
हाँ अभी तो मैडम नहीं डॉक्टर ने कहा था ना मुझे उन्होंने कहा
ऑडियो को मैंने करना पे कर दिया। हेलो हेलो हेलो।
Hello, 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 check clear, hello, hello, check. Hello, hello.
and the head of Department of Pathology at Pak Emirate Military Hospital, Rawalpindi. Sir, I would like to invite you to take this seat. Ji, sir.
I would like to introduce our co-chair for the session, Dr. Seema Irfan. Dr. Seema is currently serving as Professor of Clinical Microbiology at Aga Khan University Hospital in Department of Pathology and Laboratory Medicine. Dr. Seema has special interest in infection control, and to date she has traced many outbreaks, both in ICUs and NICUs in the hospital setting. Dr. Seema, please have a seat in front. Thank you, Dr. Seema. Thank you, Dr. Irfan. Due to some logistic issues, we have actually changed a little order of presentations. As our second speaker, Natasha Griffith, she is online right now and at Georgia, America. So it is too late for her. So we thought that we can put her talk first. It is my pleasure to introduce Natasha Griffith. Natasha has a wide experience of biosafety and biosecurity. She's currently directing the containment laboratories at State University, Georgia. Previously, she has also worked in the same capacity at Center of Disease Control and University of California, Los Angeles. So without further wait, over to Natasha.
Hello, everybody, and thank you very much for inviting me to present at this very important conference. I apologize for not being able to be there in person, but I hope that I will be able to share some of my experiences with all of you. Before we proceed, I just want to make sure that you can hear me and also that you can see my screen.
Yes, Natasha, we can see you and we can hear you. Good to see you.
All right, can I share my screen with you? I think I asked for permission.
Yes, please.
You have to let me do it. I think it's on your end. Thank you.
अपनी end से करेंगे.
Okay.
Can you do it now, Natasha?
Okay, I'm going to try. Okay, can you see it?
Yes, we can see it.
Okay, let me just try to put it in presentation mode. It's really good. Go. All right, is it in? Okay, I think now it's good. All right, perfect. Thank you very much again for having me participate in this very important conference. And again, my apologies for not being able to join you in person. So today I'm going to talk about prevention, investigation, and management of infectious disease outbreaks in a laboratory setting. So I would like to start with a few definitions. In the interest of time, I'm not going to read these definitions, but I will share my presentations with all of you so you can go through this. There's a couple of very important concepts that I would like to emphasize. There is sometimes a confusion when we talk about infection control or infection prevention control and biosafety. Those are similar, but however, they are not the same. What do I mean by that? When we talk about infection control, we talk about policies and procedures that are used to minimize the risk of spreading infections in a hospital's and human or animal healthcare facility. So it's a very hospital related. Versus biosafety, which is containment principles, technologies, and practices that are implemented to prevent the unintentional exposure to pathogens, toxins, or their accidental release. This is more in a laboratory setting. What I would like to remind everybody is that both of these rely on risk assessment and risk management process. And I will talk a little bit more about that. So when we talk about the process, we obviously want to prevent spread of any infectious disease, be it in a hospital setting or in a laboratory setting. So in a hospital settings, a lot of times we will talk about standard precautions that will help prevent infection and spread of potentially infection in a hospital settings. Some of these strategies will include hand hygiene, personal protective equipment, cleaning, appropriate handling and disposal of sharps. In a laboratory setting, when we talk about prevention, we talk about risk assessment-based mitigation strategies that usually include engineering controls, administrative controls, policies and procedures, as well as personal protective equipment. In case of an outbreak, we proceed with investigation. And for those of you that did or are still doing epidemiological in outbreak investigation, this should sound familiar to you. I did make it a little bit more concise, but basically it consists of determining if an outbreak is truly occurring, then defining and identifying different cases, developing a hypothesis, then you evaluate that hypothesis and perform potentially some additional studies. Then we put in place appropriate mitigation strategies. When we talk about outbreak investigation from the laboratory standpoint, specifically clinical laboratory, we have to put in place appropriate mitigation strategies right from the beginning, because the goal here is to prevent spread of any potential infection in that laboratory setting. We want to conserve and empower our workforce to be able to continue to do the tests and provide information. Then we're gonna advise on proper materials, procedures for collection, storage, and transport of those samples that are collected in the field. Establish policies and procedures for testing, and very important, timely reporting of results from those tests. And of course, we continue to monitor and surveil and report the tests that we come in contact with. Both of these converge when it comes to communication. A lot of times what I've seen in my experience that there is sometimes a break in communication between outbreak investigation in the field and laboratory testing. So it's a very, very important that that communication is there and it goes both ways, from the field to the lab and from the lab to the field in a timely manner. And of course, once those mitigation strategies are put in place, both in a field as well as in a laboratory, we need to be able to evaluate effectiveness of those mitigation strategies. Then moving on to the management, when we talk about, again, hospital settings, we're going to talk about universal precautions, standard precautions, transmission-based precaution, and sometimes other based on the outbreak that we are dealing with and a mode of transmission. In a laboratory setting, we're going to talk about laboratory-specific precautions, and these are based on a comprehensive risk assessment, and we'll see a little bit more what that means. Before I proceed with the concept of risk assessment, I would like just to address some common misconceptions. Infection prevention does not equal biosafety. Also, hazards that do not equal risks, we'll see that. Risk groups do not equal risk assessment, and risk groups also do not equal biosafety levels. So let's talk a little bit about hazard versus risk. Very briefly, hazard is a potential source of harm. That can be a biological, chemical, radiological, physical, anything that can harm a person. This is especially important in a diagnostic laboratory because we deal with different samples that can be biological for the most part, But we also have chemicals in the lab as a part of the reagents. We also have instruments and machines that we're going to operate for testing. So there's a lot of different things that are happening. So it's important to know what hazards we are dealing with. On the other side, when we talk about risk, risk really is probability of an event happening and then a consequence of that event. So probability will address factors that will affect whether or not an exposure will happen. So something, a break in a process. Consequence will address factors that will affect severity of an exposure event. So if we do get an exposure, especially if it's in a laboratory setting, especially if it's in a case of an outbreak, what is the severity of that exposure that can lead to potential infection and potential disease, and what other consequences could be associated with that. So again, hazard is something that is dangerous, so that danger we can encounter and can be in a lab or in a field. A risk is a function of the probability and a consequence of an event occurring. So again, hazard does not equal risk. So here's an example, and really briefly, to illustrate what I mean by this. So imagine that you are in an open field next to a very hungry, aggressive tiger. The tiger is not restrained and sees you as a food. Where do you think, what is the likelihood of an event of that tiger attacking you? And then what is the consequence of that? Both are high. Likelihood of that tiger being able to attack you is high. And a consequence, meaning you being eaten by a tiger in this particular scenario, is also high. So this risk would be considered unacceptable. Now let's look at the next scenario. You are in a zoo, you are watching a tiger that's in a cage. The tiger is well fed and it's not very aggressive. So what is the likelihood of that tiger attacking you? Probably very small. What is the consequence if for some reason the tiger would break out of the cage and attack you, of you being eaten by that tiger? Well, that might still be high. So if we look at what happened is the likelihood in this case is very low, but the consequence is still the same. And this is something in a laboratory setting that we need to understand. Just because the likelihood of something happening is low, the consequence might still be high, especially if we are dealing with an outbreak of an infectious disease that we know very little about. Very, very often this happens in the beginning of an outbreak. So the third scenario, if you look at that you are holding a little tiger, that it's a cub tiger, and it has a very playful temperament, but it's in your arms. What is the likelihood of that tiger attacking you in some way? Well, probably pretty high. But what is the likelihood what is a consequence of that tiger potentially eating you? Well, that's low. It's a little tiny cub tiger. So in this case, we have a consequence. It's a something. Think of something as an attenuated strain and something not as maybe a high mortality or in an infectious disease situation. So this is just to illustrate how we look at the risk. Let's look in a laboratory setting. So again, if we look at the hazards, we have different hazards. I'm just taking some examples. For example, if we have sharps that are dirty and that we used to draw blood or something like that, what would be a risk? So we have a hazard, which is a dirty sharp. Risk is potential needle stick and potential infection from that exposure if we are dealing with a dirty sharp. Hazardous chemical is another hazard. Risk is potentially that chemical splashing or vapors that might cause some adverse effect. Another hazard might be a biological agent. We are talking here about infectious disease. So again, it could be some type of accidental exposure or sometimes unintentional release. We talked about this, so I'm going to skip it. So let's talk a little bit more about risk assessment. So really it's a process that characterizes a particular situation in order to determine the likelihood and consequence of certain adverse effects, and then supports decision-making regarding the acceptability of the risk. So how do we evaluate that risk? So to illustrate that, I'm going to use an example, a very simple example of crossing the street. We all cross the street. Doesn't matter where we come from and which city we live in. We have to cross the street, and we cross the street many, many times. So here are the steps when we talk about risk assessment. We like to use the example of the crossing the street. When we want to cross the street, we're going to identify the hazards. So what can hurt us? Is it the car? Is it the truck? Is it the bicycle? Is it the motorcycle? Once we identify hazards, we are going to look at the risks. So we're now going to assess how fast the car, truck, bicycle, motorcycle is coming. Is this a big road that we have to cross multiple lanes, or is this a small road? Is there a stop sign or a stoplight that we can wait before crossing? Are we wearing high heels or tennis shoes? Do we have a child with us? And so we are going through that risk assessment process. Then we're going to characterize the risk. What can go wrong? Basically, what can hurt us the most? Is it the car that's going really fast, or is it the big truck, big bus? And then when we're going to prioritize the risk, so we know what are the highest risks that we are going to run into if we decide to cross the street. After that, we're going to try to manage the risk. So what controls can we use in order to manage the risk of crossing the street? Like I said, are we going to wait where is a crosswalk, or are we going to stop, wait for a signal of some sort to turn on? Are we going to get a better running shoes? Are we going to choose to cross the street somewhere when it's maybe fewer lanes? And then, of course, we determine, last but not least, we're going to determine, based on all of this and our management strategies, is that risk acceptable to us? Do we still want to cross that street? And of course, if we have somebody with us, we need to make sure that we communicate our intent of crossing the street to whoever is with us. So this is a very simple process. And the reason I'm using crossing the street is a lot of times when we talk about risk assessment, people get a little worried and talk about, well, it's a complicated process and I don't know if I can do it. Yet we do the risk assessment process daily for other things in our lives. So this is just translating it into our work environment and into our laboratory setting. So risk assessment will take into account, like I said, likelihood of an event happening, the consequence of that event. It's going to look at the people performing the tests. How is their competency? How well they were trained? How well their health status, and not just physical health, but emotional. Are they stressed? Are they overworked? Do they have everything they need to conduct testing? And then, of course, their behavior especially during emergency situations when we are dealing with an outbreak. Also keep in mind that a lot of us perceive risk different. So keep that in mind, that always plays a role. Also, risk assessment is a team effort. So it's never just one person performing risk assessment. We come from different backgrounds. We have different experiences. We also have different tolerance for that risk. So doing risk assessment as a team, you will get the best results. And it's also an ongoing process. Like I said, every time you cross the street, you're going to go through that risk assessment process. It's not, you don't do a risk assessment only once, first time you ever cross the street, and never again when you cross the street. So this is same for laboratory setting. You might need to do these risk assessments each time you get a new sample, if there's a suspected new outbreak, or anything along those lines. New reagents, new machines. Once we do risk assessment, we are moving into a risk management. So risk management really is the process of defining mitigation strategies, plans, controls, on how to reduce the risk to an acceptable level. So this is a very complicated schematic here that I'm showing you, and it's just one of the risk management approaches. So I'm not going to go into the details, but the idea is to show you that you can develop a flow chart or a tree on how to manage risks in your work environment, in your laboratory. And so this specific example talks about assessment mitigation, which I talk about, but also performance. How do you verify that your mitigation strategies that you have put in place are actually working, that they are sufficient to protect your staff and people in the laboratory and outside? So we talked about risk assessment. Like I said, for risk mitigation, there are many options. If you are in a clinical laboratory setting, a lot of times you'll talk about, when people talk about risk mitigation, they often talk about elimination and substitution first. What I want to emphasize is when you are in a clinical setting, you don't always have an option of elimination and substitution. You never know what's going to walk through that door, what sample is going to come in, and what's in that sample. So we're going to rely a little bit more heavier on other controls, such as your safety equipment, including your personal protective equipment, your management and how you perform the certain task, your training of the staff, how they are trained, as well as your work practices. So you want to have a good work practice, especially and you want to practice those work practices, especially before an outbreak situation happens. And last but not least, you want to make sure that you can assess the performance and how those mitigation strategies that you are putting in place are working to basically protect and mitigate or reduce those risks in your work environment. So in summary, in order to prevent and manage infectious disease outbreaks, especially in a laboratory setting, we need to be able to conduct a comprehensive risk assessment and implement effective risk mitigation strategies. We talk about that this is an ongoing process, and it will take into account multiple factors such as agents, procedures, activities, people, environment, all different components. Also, risk management is a continuous process as well that will enable us to reduce risk to an acceptable level. But please remember that risk will never be zero. You don't want to become complacent and say, well, there's no risk, therefore I can relax. I don't have to worry about, this is just my usual routine. I know what I'm doing. That's where potential exposure can happen because you become complacent. So please remember that risk is never a zero. And last but not least, laboratories, clinical laboratories, play a critical role in an outbreak investigation. So I will stop here and open up for any questions. Thank you very much.
Thank you so much, Natasha. Can you hear me now? Okay. Natasha, thank you so much for giving us your time and for such an informative talk. Can we take just one question, because Natasha won't be here for the panel discussion, if anyone have? I think we were not ready for the questions at the moment. Natasha, thank you so much. I'm honored to be your mentee and you being my mentor. I still remember the first day of my fellowship with Natasha at University of California, Los Angeles. And the first thing she told me was her first slide, that look, Sana, this is not infection control. This is biosafety and biosecurity. So thank you, Natasha. Thank you so much.
After this wonderful talk, we have the honor to have Dr. Afia with us. Dr. Afia does not need introduction in the microbiology community, as most of us know her as a colleague or as a teacher or as an examiner. Dr. Afia has more than 25-year experience in the field of microbiology and hospital infection control, and she's working as consultant microbiologist at Aga Khan University Hospital. Madam, please, over to you.
मामाए नहीं भाई बहुत सारे हैं और भी हाँ स्लाइड लगाएंगे मेरे प्लीज हाँ और पीछे जाएं thank you Assalamu alaikum. First of all, thank you very much organizing committee that they provided me chance to discuss this important topic with you all, and I will start from where the last line of Natasha's slide. Laboratory plays very important role in outbreak investigation. So what we are going to do in next 20 minutes, outbreak investigation and the good outcome. So and practical experience what we have done during last few years. So first of all, something about non-tuberculous non-tuberculous mycobacterium. Since many junior colleagues are here, so I would like to just give you some introductory remarks that they are everywhere. They are in water, they are in river, and more than 190 species have been identified and 40 are considered pathogenic. So we have environment exposure, not only in hospital. I'm going to talk about outbreak, but they are also at home. And they are organisms of low virulence, and mode of transmission can be, if we can guess, aerosol ingestion and inoculation, and in incubation period from weeks to years. So that's the reason many times we miss that, and it depends upon the type of NTM. So very nice classification by Runyon, and we all know this classification. There are 4 groups, and they characterize it on the rate of growth, especially the formation of pigments in the light, in the dark, or no pigment, and the rapid grower. The important thing is time of growth. So we can see that they grow a variable rate, and frequency of nosocomial infection. I will not go in the detail of name because that will confuse all of us. So it's just NTM at the moment. So virulence factor. Thick waxy wall and presence of libo-arabinomannan, they modulate our immune system if they are there. And very important thing is production of biofilm. Someone was asking this question in the morning, that production of biofilm. So this is the organism uses this characteristic, and it grow well on non-biologic product, and they form biofilm, they multiply there, and they have their, Dr. Irfan has described in the morning, and then they are there in the pipes, in water, in medical equipment, and their tunnels. So, and very nicely, one of the editor of this journal, Microorganism, this year we have a full supplement on NTM. So very nicely he described that why we are having more and more NTM infection in the hospital. And he is saying that we have created ideal habitats for the NTM, particularly via plumbing. Why? Because what plumbing does, it surfaces that promote adherence and biofilm formation. Disinfection, the routine disinfection kills other competitors, but it survives. Heating, which increases the number of NTM, and stagnation, under which NTM grow happily. So this is the scenario we see in the hospital. So in community, broad range of infection, we can see that there are many types of infection, and in hospital where the number of nosocomial infections are on rise. So they are post-surgical, especially prosthetic surgery, whether it's a cardiothoracic or any type. If there is a graft, if after bronchoscopy and peritoneal dialysis is skin. So you can see that almost every organism can affect if it is there. So this is the nosocomial infection. And then what are the sources? The sources almost always linked with something to do with the water process in the hospital or in the system where we are using them. So they are syringe, they are solution, they are water, and they are injections. So there are so many things. So when I looked for the literature, I found that during last 2 decades, there are many papers internationally that they have, and there are many outbreaks which can be laparoscopic surgery, which can be a spinal surgery, eye surgery, anything, they are there if there is some foreign body. Then I looked for the Pakistani literature. I am happy to tell that there are Dr. Imran group with Dr. Rumina Hassan, they published 2 articles about the rise of this organism. This is from the right-hand side, this is from the laboratory, and the left side is the one which is one of the clinician. He published article about the total knee replacement and the presence of NTM in their wounds, which is alarming. So then we should not forget community, because cosmetic surgery, acupuncture, piercing, very small things like tattooing, like Botox toxin, all of them, they can result in infection which is the subcutaneous infection or systemic infection, and very difficult to treat. This is the problem. And thanks to Dr. Bushra Jamil, I was speaking with her yesterday, so she told me that she has a patient, young energetic patient, and she decided that she will have some fat transfer, autologous transplant from abdominal wall to her face and to her knuckles, and the result is that she has developed NTM, and the last slide, after 6 months of therapy, she is slightly better, but still there is oozing from her face and from the abdominal wound. So with this background, I would like to slightly move over. Sorry. With this background, I would like to share few outbreaks which we handled. Outbreak 1 and outbreak 2, the first one was, we received few samples from a center. The liposuction was in their practice, and we received few samples from some people, and then there was growth of NTM. The second one was from hair transplant center. Again, there was growth of NTM, but unfortunately we could not follow these 2 outbreaks. But this third outbreak, we received few samples from a center, and the history was wound and joint infections after prosthetic joint surgery. Multiple patients experienced this problem, and swab and pus were received in the laboratory, and we grew organism. We were in discussion with the consultants, and we investigated this outbreak. You can see that in this slide, we did surveillance culture multiple times. For example, washing area 7 times, washing sink and washing machine, so RO water, overhead tank, drill machine. There were so many things. And you can see that by our phenotypic method, we identified multiple types of micro-NTM. And unfortunately, because of the non-availability of sequencer at that time, we could not type them. But it tells that there was bioburden. There were too many organisms around. So, and the second outbreak, very recent one, we grew NTM in blood culture from a center after cardiothoracic surgery. We received blood culture. 11 children blood culture were positive after heart surgery. And surgery was done during last 3 to 4 months, and the age was 2 to 15 years. Most of them, they were with congenital heart disease, and the type of surgery is written here. And later on, after the sensitization, from that center, we also received blood from adults, and 2 blood culture adults were positive. So what we did, we received the sample, we grew them with the help of various media, and we did the MIC, and we tried our best by phenotypic method to identify them. So for investigation, we visited the site, their OR, their CSD, RO plant, independent interview of surgeons, OR technologists, CSSD, and MEP staff. QC records were checked. They were available, not available, incomplete. So many problems were found and highlighted, and we took surveillance culture from them, and it was submitted to us. So this is the scenario. This is cardiothoracic unit somewhere in Pakistan, and actually the water supply was tube well, and so we checked water at tube well entry site, main tank, scrub area. So there are every possible place, and you can see on that column that heating cooling unit OR 1, 2, 3. We found we found NTM in those samples. So what is heating cooling unit? I would like to explain to you just in a minute. Whenever they do surgery, open heart surgery, the heart lung machine is an essential part of that surgery, and heater cooler unit is also there. So what we have to do, surgeons, what they have to do, they have to have a system where patient's blood should remain at 37 degrees centigrade. So they use heart-lung machine. What blood comes from the superior and inferior vena cava, blood comes to the machine, and the oxygenation took place. But to maintain the temperature, it goes into another chamber, which is warm chamber, and but the blood is in the tube. It is in a water tank, and the second one is a cold chamber, and this is to maintain amount of blood or cardioplegic solution, because they want heart in diastolic, which should remain cold, and in diastole. So this is one circle which goes to heart, and this is the second one. The other important thing is that to keep the circulation of, to maintain the heat, what they do, they use fan just in the bottom of the machine, and this just disseminates air here and there. But then what happens, this heat cooling system, they are not airtight, and they have fan. So what happens, if the machine or if the water in the water tank, which is not airtight, if it is contaminated, it can spread organism in the environment. And this actually can happen if there is a cardiothoracic surgery. So if the water is contaminated, then the aerosol are here and there, and they will directly lodge into the wound. And if operation is usually 4 to 5 hours, and this can contaminate the instrument or the prosthesis, anything. So yes, this study was done in Switzerland. Actually, what they did, they very smartly, what they did, just concentrate on this column, the right one. What they did, they in an empty OR, they used a heart-lung machine and the one which used the fan. And what they saw, they did a smoke test, and they found that whenever there is a machine is on, the smoke is there, and it just spreads in the room. So what they did, they took they just changed the face of the machine towards the exhaust fan. So on the left side, bottom one, you can see that when the exhaust fan of the operation room is on, the whole the smoke shifted to that side. So it means that it is not spreading the organism in the same room. So actually, this was the first study which actually clarified that NTM spreads in the OR with the help of aerosol. It is not only the contaminated instrument, but it can be aerosol. And very interesting thing that from 2012 to, there was an organism, a rare organism, Mycobacterium camerae, a rare pathogen. This was found in 3 continents in the world, America, Canada, New Zealand, Australia, and Europe, that it was causing post-cardiothoracic infection among patients. And I think 168 cases were reported. And that was, very interestingly, that was just related with a particular machine that was manufactured in one of the country. And very interesting, finally they found that those machines and their tubes were contaminated at the source. It means in the company. So anyway, that is one thing which is reported in literature, and again due to the presence of this machine. But our problem was slightly different because we have variety. And the organism were there. So one of the recommendation is that why don't we keep these machines outside the room? But technically this is difficult, but in Switzerland, scientists or one of the hospital, they developed OR, and the 4 OR with a small room where they kept this machine, which is outside. So now they don't have any NTM infection in their institute. So this is one of the interesting thing, that this machine is there, so this is always a hazard for all of us. Now the challenges what we face in the laboratory and the clinician when they treat patient. Challenges for us, for both parties, there is a challenge because the organism, it grow very slow, and we from the laboratory side, I must say that usually we don't receive any history. When we don't receive any history, we don't look for this organism, but for the clinician and us, our index of suspicion should remain very high. So in the laboratory, the usual practice, we keep our plate for 48 to 72 hours, and then we discard them. We don't look for NTM. They don't grow in 48 hours. They grow after, even rapid grower, they grow from 3rd day to 7th day. So for clinician, we request that please inform lab, and it's important if you also send TB culture. And these organisms, when they are present in the clinical specimen, at times they don't take FB stain. This is another problem what we face in clinical side. And ID and susceptibility testing are difficult for any ordinary lab. And for outbreak strain, molecular tests are now we have acquired in Pakistan at few places, but very expensive. So people, they don't go for that. So message for my laboratory colleagues: in case of suspicion of growth is there, make a smear in saline. If there is a growth, try to make a saline. Actually, we say that it is Imran's technique. Our colleague, Dr. Imran, actually, he taught me this. Okay, if there is a NTM, Dr. Afia, just put it on the slide, make a smear, it will float. It does float. So we say that it is Imran's technique. So this is very useful, and this is very useful. And then for the to convince organism to behave and take FB stain, if we grow them on 7H10, then they take. And during this outbreak and the previous one, 3rd and 4th, actually we experience all these things. And check vancomycin susceptibility from gram-positive rods on the plate, and then we will find that NTM are resistant to vancomycin. So these are the clue, and they help us. Clinician and the hospital, very difficult clinical diagnosis. Usually non-healing wound with conventional antibiotic. Laboratory reports are not supportive. I must say that many times we don't grow, so we don't support them. We say NBPI or no growth. And environmental monitoring is very difficult for most of the hospital, and it's difficult to maintain good quality water in the hospital. So management, what we did, immediate recommendations. Dr. Seema was the boss, and she said stop all device-related surgeries. We collected data from there, and we looked for the source with the aim to take some corrective measure, and we sent sample to lab, and you have seen the result. And these 4 things are very important as far as NTM outbreak is concerned. Sterilization of critical items. We found that many items, they were critical, but they were not doing sterilization properly. Quality control of high-level disinfectant. Again, at most places it is compromised. Environmental control, cleaning and disinfection of HCU is very important, and disinfection and quality control of water. So very briefly, I will go fast for these 4 because we all know that we should do this. Critical item, sterilize. Their quality control is a long list, but we have to follow. It's expensive, but we have to do it. High-level disinfection, we all know that, glutaraldehyde or paracetic acid, and appropriate concentration. And for the HCU device, don't use tap water. This is the practice to refill it. Just use tap water, only sterile water, and use less than, and if not sterile water, then it should pass through a membrane filter, which is with very small size, and direct the exhaust vents away, because it's very difficult to develop a new OR. Strict adherence to cleaning, disinfection, develop SOPs, and test environment and air culture if you suspect that. Quality control of water. So what we did actually in both places, we asked them to flush their all hospital line with the help of very concentrated, which is 10 milligram parts per million of 10 milligrams per liter of chlorine. So this is known as flushing activity. So we have to flush it, and then after that, and if someone can do it, use auto dosing system in the hospital, or if not, then add manually. And then chlorine level and maintenance, it is again very crucial thing, and we must abide that. So in summary, NTM infection are on rise. Clinicians and labs should remain vigilant when they handle an acute negative culture negative healing wound, not healing wound, and clinicians are concerned about that. Micro labs should develop expertise and hospital and cosmetic industry, because many cases they are coming from the cosmetic industry. So we should be vigilant. Sterilization, और आप सब लोग तो ज़रूर पार्लर जाते होंगे, तो वो तो serious बात है. And cleaning and maintenance of equipment and instruments should be strict. मैंने वैसे जाना छोड़ दिया है. So cleaning and okay, cleaning and maintenance of equipment and instruments should be strictly disinfected or sterilized as per manufacturer recommendations. In the end, I would like to acknowledge the effort of Seema, Javeria, Imran. Mehreen is our resident. हमने उसको line listing पे लगा दिया था. Mr. Israr Ahmed, he is our senior tech. He is a wonderful person. He does environmental culture. And microbiology lab staff, they worked with us and they tried to. So that was it. Thank you. Thank you. Thank you.
Thank you very much, Dr. Afia, for a wonderful talk. Now I would like to invite our next prestigious speaker, Dr. Bushra Jamil. Dr. Bushra is a consultant infectious diseases at Aga Khan University Hospital. She is also president of Medical Microbiology and Infectious Disease Society of Pakistan. And she has been working in the recent floods. She has been working in outpatient clinics for the flood victims. So her talk will be focusing on the infectious diseases in the flood victims. Madam, please.
Thank you very much. Assalamu alaikum, everyone. Where is my presentation? Okay, so I was given this topic of infectious diseases outbreaks in flood, ground reality. So I love to talk about ground realities. So I'm going to talk a little bit about the background context, environmental factors, infections which are endemic in Pakistan, what happened in 2022, and summarize it. So what happened this year was we had the worst floods in the last 50 years. So 1/3 of Pakistan was submerged in water, and monsoon season was the worst since 1961. So very heavy rains and the country was submerged. So this is not the first time Pakistan has experienced floods. We've had floods in the 60s, in early 70s, then 2010, and then now. But no steps were taken to prevent or be ready to meet the challenges. So during floods, what is expected is that there is direct injury from floating objects, so injury and death from the crisis itself. Then indirect adverse effects, which are brought about by forced displacement, lack of legal protection, and decreased access to healthcare. Then overcrowding, inadequate shelter, poor sanitation, insufficient water quantity and quality, reduced food security, increased risk of malnutrition, and outbreak of communicable diseases. And these were all seen during these recent floods because the camps which were created just provided shelter, and there were no wash facilities, no toilet facilities, and food which was being distributed was also not, did not meet the nutritional requirements. And extreme stressors can also trigger mental health conditions, and these somehow go unnoticed and unaddressed with all the other various huge, humongous problems which go on at the same time. And then interrupted medical supplies and interrupted healthcare systems and displaced populations also affect maternal health care and treatment of other chronic diseases which are required to be carried out regularly. So one problem was that many of the hardest hit areas due to flood were amongst the ones which were identified as the most vulnerable in Pakistan. And actually children have suffered disproportionately as compared to the adult population, as they were already suffering from malnutrition and poor access to water and sanitation, and then they were displaced as well. And the primary goal of response during the crisis was to prevent and reduce the excess mortality and morbidity, because all this was expected. To me, effective approach would be simple, targeted, relevant, and result-oriented. I'm not sure about the result orientedness of the strategy, but this is how it should be carried out. So because of a successful example of NCOC, this National Flood Response and Coordination Center was set up on the same premises, and the national coordinator from the military side was also the same. But if we compare this response to the COVID response, it was quite different because health experts were not included or engaged in the response this time round as they were with the COVID. So we used to get these regular updates from government of Sindh because Balochistan and Sindh were the worst affected areas. And according to these, the common problems were acute respiratory diseases, diarrheal diseases, malaria, skin infections, and then snake and dog bites as well. So the question arose, why were Pakistan's floods so extreme this year? Pakistan has had problems in the past as well, but this time round, the river banks broke, there were formation of new glacial lakes which burst, and this was the worst flooding event of the last century, and a huge number of people were displaced. So the main reason is, one of the major reasons actually, is climate change. So overall, globally, there has been a rise of 2 degrees Fahrenheit in the 20th century. And it is expected that this change will continue throughout this century. And this has led to shorter frost seasons. Frost-free season has extended with increased CO2 emissions. There has been increased industrialization and deforestation, all contributing to higher CO2 content, leading to global warming. There has been a change in precipitation patterns leading to more rainfall and floods in some parts of the world, and droughts and more heat waves in other parts of the world. So extremes of weather condition. It is expected that hurricanes will become stronger and more intense, and sea level will rise by 1 to 8 feet by the next century. So very, very alarming figures. So according to modeling, what we see here is an upward trend in both temperature and precipitation as time goes by. Until the next, till we arrive at the next century, I don't know whether Pakistan will be inhabitable or not. Yeah. So due to rising temperature, as I mentioned, glaciers are melting, and there are more GLOF events, which are glacial lake outburst flooding events. And these have increased. There have been thousands of GLOF events, which is very alarming, and nobody seems to talk about it. So malaria and dengue have both moved to higher altitudes in northern areas because the temperature is high and situation is more conducive for local mosquito breeding, which was not seen before. And again, rainfall and post-flooding situation and garbage, and they've all encouraged vector breeding and disease spread. So what happens is that at higher temperatures, larvae take shorter time to mature, and the biting frequency of arthropods increases, and there is, the population is amplified greatly with rise in temperature. So bite frequency of all arthropods increases. So let's take a look at what happened in the floods of 2010. At that time, obviously, gastroenteritis and cholera, respiratory infections, malaria, typhoid, hepatitis, all spread very rapidly in the affected areas. But at the same time, the mosquitoes became resistant to the commonly used insecticides. So resistance was noted. So what happened subsequently? We had torrential rains in 2019, followed by this kind of a situation. And this coincided with Eid al-Adha, and this is when we saw a surge in cases of listeria. This is in addition to the usual gastroenteritis and other problems we see. So there is a model of rise in infections which have been proposed that during floods, during the first few days, waterborne and respiratory infections increase, followed by rodent-borne infections, and lastly by vector-borne infections, because there is more water available for vector breeding. But in our setup, because we have many conditions which are already endemic, we did not necessarily follow this timeline. So strangely, cholera cases had started rising since January, which was way before the rains. And dengue cases were also rising before the floods. So if conditions are endemic, which we may not necessarily see this pattern, but conditions are amplified because of conducive environment and a lot of water for mosquito breeding. Then there is this issue of food safety and contaminated water and malnutrition. So obviously food safety is particularly important for infants and pregnant women who are most susceptible. And in the camps we conducted and attended, malnutrition was very common. We saw stunted babies and they were already malnourished. So it cannot be said that it happened as a consequence of floods, but they were malnourished to begin with. And they suffered most because of the camp conditions. And then in most of the camps, the food which was being served was rice-based and obviously was not catered to the nutritional requirements of the people who were residing there. And this was this aggravated malnutrition, but there is no system of surveillance or follow-up, so we do not know what happened to the children we saw in the camps. Then respiratory infections are common because they spread in overcrowded shelters, and they were possibly viral, a large majority of them. So if we had baseline data, we could have predicted that which ones would be prevalent, and we may have been in a position to do something about it, but the data was missing. Then cholera. So cholera is, outbreaks of cholera are associated with rise in temperature of ocean water because cholera resides in phytoplanktons. So I think this was the reason why we saw cholera even before the floods. Using artificial intelligence, it has been shown that cholera cases are associated with periods of low precipitation and high temperatures and not actually with floods. What floods do is they just disrupt the water and sanitation system, lead to more contamination. So natural disasters are contributory in that manner. But cholera cases started appearing months before the rains, and the number of cases went down actually with the onset of floods, which you can see in this slide. So the lowest one shows the trends of cholera. So highest in the earlier months, and then dysentery and non-cholera diarrhea were the ones which rose with floods. Now, this picture here shows administration of cholera vaccine by Dr. Palita. So for Pakistan, there is a mismatch between problem and the strategies which are proposed or are practical and can be implemented. So instead of ensuring clean water, the only option which is often available to us is that of vaccine. And in fact, it was suggested that cholera vaccine be administered to all the IDPs and in the flood camps. But luckily, the cases were not that many and we did not see a surge. Moving on to vector-borne conditions, burden of malaria and dengue increased as expected, but again, dengue had started increasing even before the rains. So it is endemic to Pakistan. We see cases the year round. But by mid-June, the number of cases was significantly higher if you compare them with the previous 4 years. So floods did have a significant impact on the total number of dengue cases and it spread. So dengue was rampant because of logarithmic increase in mosquito breeding. Mortality is low, but the higher the number of cases, the more severe cases you see and the more people die. This is just a map showing dengue. For malaria, again, we saw an unprecedented increase in malaria this year and a huge increase in cases. So this year, around, till August, there were 3.4 million cases compared to 2.6 million cases last year during the same time period. Large majority, 73% were vivax and 23% were due to Plasmodium falciparum. So, and the worst hit districts were in Balochistan and Sindh. Again, there was mass distribution campaign for long-lasting insecticide impregnated nets. You cannot trap or enter someone in a mosquito net when the mosquito, especially in the context of dengue, can be a day-biting mosquito. So again, a mismatch between the problem and the strategy which was used. So a lot of funds were diverted towards procuring and distributing LLINs, which is a suboptimal strategy. Then again, chemoprophylaxis on large scale was suggested, but it's not a recommended strategy. And fogging is something, insecticide spraying is something which appears to be very attractive and first thing which we all go for. And in fact, this is a suboptimal strategy because then insecticide gets diluted out and mosquitoes are not controlled. And then this the larvicidal strategy. The government apparently purchased huge quantities of larvicide, but whether it was used or not, we have no information. Then residual insecticide sprays, I don't know whether this strategy was used or not. And lastly, rodent-borne diseases. We don't think about rodents as a big issue because we don't think about it, we don't have any diagnostic tests, and then we forget. So this is something which has been suggested as a major issue after floods because the rat excreta actually contaminates your water supply. So just to summarize, I've written here, added fungal and bacterial skin infections, scabies, and head lice. So these were quite common in the camp situation and had to be addressed because I think almost everyone was suffering from some kind of skin infection or the other in the camps. That was in addition to all this. So to summarize, surveillance, outbreak detection, and early response are essential. Our response this time round was based on voluntary medical help and other relief camps. They were largely set up by people who just volunteered. Following floods, we know that respiratory and gastrointestinal infections are common early on, followed by vector-borne diseases because of post-flooding water stagnation. Management decisions have to be based, had to be based this time round on clinical diagnosis because there was limited access to on-site diagnostic tests. And lastly, there was a mismatch between problems and mitigation strategies and no concerted effort or planning. We have not been engaged in any such activity for preventing or dealing with such disasters in the future. Thank you very much.
Thank you so much, Dr. Bushra. As we are sitting today here, and I'm sure that we all will raise the hand that we have witnessed one pandemic, and that is COVID-19. But before that, there had been many, many pandemics in the world. In another 10 minutes, I'll take you to a short recall or a flashback of all the pandemics that this world has ever witnessed. As a microbiologist, I know we have a passion that we love to see our small micro world through the lens of a microscope. But now I'll take you through the lens of a camera. Please have a look at it. Thank you.
AIDS, acquired immune deficiency syndrome. No one with AIDS has been cured.
AIDS is now the leading cause of death among young people in this country.
that COVID-19 can be characterized as a pandemic.
Absolute constant, this killer pandemic is virtually out of control. The patients are literally gasping for air. Nobody expected this. Nobody thought they'd be treating so many so quickly. And it's not like flu. It is chronic pneumonia, and it is killing hundreds each day.
In China, the city of Wuhan is still on lockdown, with Americans trapped inside as the number of worldwide cases climbs to more than 2,000.
It is somehow unimaginable, unbelievable, that in the most famous city of the richest country in the world, they're digging mass graves for those who can't afford a funeral and for those who died of coronavirus with no next of kin.
Have gone viral on social media. My colleague Maya Sharma joining us with the latest. Maya, we can see shots on our screens at the moment of a group of men in hazmat suits dumping body bags into a huge pit. What really is going on?
चौबीस घंटों में 5,870 नए केसेस रिपोर्ट, मुस्बत केसेस की शरह 10.9 रही।
So this is reality. This is reality. Okay. So pandemics are not new to us. Pandemic had been there, and I think as healthcare professionals, as we advocate the policies and a system that we want to make for our country, I think we have to learn from the mistakes that we have done in the past. With this, we will close our session for the talks. And do we have time for panel discussion now? Yeah, if there are any questions and answers, we have the panel of spec. They are after the lunch. Yeah. So if there are any questions from Dr. Afia, Dr. Bushra, Natasha is not here with us. Safia, now you have no question from Dr. Afia. माइक दे दे प्लीज।
जो आपने एनटीएम का जो बताया है, ये I actually don't need मतलब you have isolated वो जो आउटब्रेक में different NTMs actually different species of NTM और वो लिंक हो गई थी सारी पेशेंट के साथ स्पीशीज टाइप, जिस तरह जो हमारा क्लासिकल वे होता है कि सेम स्ट्रेन्स या अपने ओवरवेलमिंग एविडेंस ये था।
हम लोग के पास ये ओवरवेलमिंग एविडेंस था, हम लोग उसका वो मॉलिक्यूलर टाइप तो नहीं कर पाए, लेकिन चूंकि उसको हम इस तरह से लिंक कर सकते हैं कि देयर वर ग्रोथ ऑफ NTM फ्रॉम पेशेंट्स क्लिनिकल सैंपल, और जो कि नहीं होने चाहिए थे, क्योंकि अच्छा मैंने जिक्र नहीं किया पूरी टॉक में लेमिनार फ्लो का। दोनों सेंटर में लेमिनार फ्लो बहुत अच्छे काम करने वाले थे, तो वो इशू नहीं है। लेकिन जो पहला वाला सेंटर था वहाँ बड़े मल्टीपल टाइप्स थे, आपने नोट किया होगा कि उसमें 3-4 तरह, तो हम पेशेंट से तो इसको कोरिलेट नहीं कर सके, बट आफ्टर ऑल प्रिवेंटिव मेजर्स केसेस आने स्टॉप हो गए हैं। देयर आर नो मोर केसेस ओवर देयर। सो एक आइसोलेट की आउटब्रेक नहीं थी उस सेंटर में, लेकिन दूसरे वाले की भी एक ही टाइप निकली तकरीबन, लेकिन हम उसको ना टाइपिंग नहीं कर सके मरीजों के साथ। हाँ हाँ स्पेशिएट कहते हैं, रैपिड ग्रोथ थे ये जो हाँ 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 हाँ
एक और मेरी
all the strains from the patient samples as well as from the environmental
fourth
cultures.
outbreak,
The last outbreak that was one of the biggest center of cardiothoracic surgery. So we identified Mycobacterium abscesses from the clinical samples as well as from the environmental sample, and that was mainly water culture. From the air handling unit. Yeah, it was. But we didn't perform the sequencing, so we can't say right now that these are the same, but in the future we will be going to do that.
What advice would you give to other labs?
Other labs?
Rapid growers को तो pick कर लेते हैं सब. From what about other types for wound infections?
Actually हाँ वही वाली बात है कि इसमें पता है रोल दोनों का है, क्लिनिशियन का भी हम लोग का भी है. इंडेक्स ऑफ सस्पिशन शुड बी वेरी हाई, नंबर वन. अगर जैसे अभी एक और आया हुआ है हमारे पास एक, उसमें उनको शक है, हमें शक है, फिर उनको भी शक, हमने डलवा दिया उनके दिमाग में भी. सैंपल आया हुआ है, हमने उसकी इंक्यूबेशन बहुत लंबी कर दी है, वो हमने प्लेटिंग डाल दी है कि 20 दिन से पहले उसको नहीं करेंगे. अभी तक उसमें कोई ग्रोथ नहीं आई, 1 हफ्ता हो गया है. तो मीडिया अच्छा जहाँ से अब आया है, दे आर स्मार्ट इनफ, उन्होंने हमें रूटीन कल्चर भी भेज दिया, टीवी कल्चर भी भेज दिया है. जो ब्लड कल्चर की प्लेट थी उसको तो हमने पूरी टेप लगा के उसको अंदर बंद कर दी. सो फार देर इज उसमें सिर्फ स्ट्रेप्टोकोकस की ग्रोथ थी, लेकिन हम चाह रहे हैं कि उसमें ग्रो हो जाए, उनका मसला हल हो जाए. तो वो अभी तक ग्रो नहीं हुआ, इंतज़ार है. तो अभी सिर्फ 5 दिन हुए हैं, ग्रोथ नहीं आई है उनकी.
And so from the clinical sample, it is not difficult because you isolate most of the time single organism from the critical sites. But the most important thing from the laboratory perspective is that when you get samples from environment, there will be lot of other organisms growing. Okay, because we took sample from the surfaces, we took sample from the water, and there were many other organisms. So because these organisms are very slow growing, even if we label them as a rapid grower, but they do take 5 to 7 days sometime. And in between the other organism, you have to identify things. So that needs very important insight, that you have to make acid fast staining of all the colony that are growing from the confluent growth, and then you have to isolate that particular isolate on some of the LJ media or any kind of 7H11, we use that one, and you have to do repeatedly subculturing and isolating things, and then it needs lot of patience. Yeah, and I would thank to Javeria, Israr Sahib, Imran, and whole of our microbiology team, that they took so much critically, they evaluated that thing, and they finally became to get that isolate. Yeah.
एक चीज़ मैं बोल दूँ इसी को कंटिन्यू करने के लिए. एक चीज़ जो है वो ये कि जब पेशेंट के पस की ग्राम स्टेन हम देखते हैं, वो थोड़ा सी फील आ जाती है, जैसे एनारोबिक की फील आती है ना, इसकी भी थोड़ी सी फील आती है कि ऑर्गेनिज्म होते हैं, डिस्टर्ब किस्म के ग्राम पॉजिटिव ऑर्गेनिज्म होते हैं, कहीं पे अजीब अजीब शक्लें होती हैं, फिर उसको देखें, ढूंढें, फिर उसको FB स्टेन करें, कभी से ज़रा सा FB स्टेन हो जाता है, फिर उसको करें. तो इट्स अ टीडियस जॉब, जैसे सीमा कह रही है कि टीडियस जॉब ज़रा करना पड़ता है, फिर वो ढूंढ के निकाल लेते हैं. एक चीज़ और कहना चाह रही हूँ कि जब ये टीबी लैब में जाता है, आप में से जो लोग टीबी लैब में काम कर रहे हैं, हम टीबी के लिए 1 मिनट, अच्छा आप कर रहे हो, तो जब हम टीबी लैब में काम करते हैं तो हम टीबी के लिए सैम्पल्स को हम डीकंटामिनेट करते हैं. ये NTM जो ज़रा हार्श तरीके से उनको हम डीकंटामिनेट कर देंगे तो फिर दे विल डाई. ये मुझे जवेरिया ने अभी मैसेज दिया कि आप ज़रा टीबी वालों को ये तो बोलिए कि हम भेज रहे हैं सैंपल, इसको ज़रा हार्श वाली नहीं करना. तो इनके भी ना थोड़े से डिफ़िकल्टीज़ हैं लेबोरेटरी में ग्रो करने के, तो बट वी हैव टू डेवलप कैपेसिटी ताकि ये ग्रो करें. जी प्लीज़.
मैम, थैंक यू वेरी मच फॉर योर एनलाइटिंग टॉक. I fully agree with you that we have to raise the index of suspicion. Otherwise, it's not possible to detect these microorganisms. What specific and sustainable infection prevention and control measures you would like to advise in a hospital setting?
Okay, 2 to 3 things very important. हम cardiothoracic छोड़ देते हैं. First of all, CSSD. Sterilization and disinfection in most of our hospitals are compromised. They don't use quality control. वो जो अपने ऑटोक्लेव के बाहर भी जो पैरामीटर्स होते हैं वो भी नहीं होते हैं, एंड वी डोंट कीप रिकॉर्ड. ये हम लोग का अलग प्रॉब्लम है कि हम डॉक्यूमेंटेशन नहीं करते हैं. सो फॉर दिस टाइप ऑफ इन्फेक्शन, इफ इट इज़ ए क्रिटिकल आइटम, इट हैज़ टू बी स्टरलाइज. If it is something for the bronchoscope, so high level disinfection is mandatory. So these two things are very important as far as these things are concerned. And if अगर कहीं पर है तो chlorination of देखिए chlorination पे बहुत ज़्यादा ही कि concentration ज़्यादा होगी तो वो पूरा hospital का pipe का system ही ख़राब हो जाएगा. तो ज़रा limit में रहकर if there is a problem then one has to do it, otherwise इसकी ज़रूरत नहीं है. But disinfection and sterilization is crucial as far as hospital practice is concerned. और जो cosmetic वाले हैं वो भी ज़रा अपने अपने parlor को देख लें कि वहाँ क्या हो रहा है.
Thank you very much, ladies and gentlemen. Now we will proceed for lunch, or after lunch there will be a free paper session of free papers, and अच्छा अभी फ्री पेपर है. नहीं लंच नहीं है, बेबी अभी लंच नहीं, फिर फ्री पेपर नहीं होंगे, ऐसे कैसे हो सकता है. सेशन यही है, लेकिन ब्रेक के बाद इसी के फ्री पेपर्स कंटिन्यू होंगे.
बस अब खाना खा के आ गए, लेकिन दिक्कत यही है कि ये बेसिकली सेशन वाइज यूनिट था. अगला सेशन अगला है. बस ऐसे ही
क्या है.
We excluded all the non-registered patients and our registered patients who were not having any central line. Central line days were assessed to determine if a laboratory confirmed bloodstream infection was really CLABSI. For that, we counted the central line days in the monthly denominator summary data. We modified the criteria to identify CLABSI cases which were not attributable to kidney center. So the cases which were attributable to our center was defined as primary, and the secondary were those whose line was either inserted or maintained at other institutes. Our results revealed 59 incidences of CLABSI in a total of 1,413 central line days. So this gave us a total of 42 CLABSI incidences per 1,000 central line days. 36 were primary cases and 23 were secondary CLABSI cases. 36 of these patients had tunneled catheters, while 23 had non-tunneled double lumen ones. Internal jugular vein was used in 50 of the cases, while femoral line was inserted in 9 cases. 37 central lines were inserted using ultrasound guidance. Out of all these 59 patients, 55 got treated, and their lines were removed and they got discharged on appropriate treatment, but 4 patients unfortunately expired, giving a mortality rate of 7% in these cases. This is a summary of this. The total number of patients with central line were 310. The device days were 1,413. 59 central line associated bloodstream infection cases were identified. 36 were attributable to kidney center and rest to the other hospitals. These were the organisms identified. Staph aureus was the predominantly isolated ones, followed by Enterobacteriales, and then Enterococcus, Acinetobacter species, Candida species, Pseudomonas aeruginosa, Stenotrophomonas, and other Pseudomonas species. Enterobacteriales generally included the predominantly identified was Enterobacter cloacae and other species. And in Candida species, we identified Candida tropicalis, Paracellosis, and some other species. In rest of the pseudospecies, Ralstonia piquetii, Chivinella putrefaciens, and Chrysobacterium species were identified. So this is a common cause of morbidity and mortality in our patients. This is a specialized hospital providing services to patients with CKD. Majority of our patients are receiving hemodialysis, for which they need either a temporary or a permanent access. When the permanent access is either not available or it is not mature enough, the patients rely on temporary vascular access, which is generally a central line which can be tunneled or non-tunneled. We have a high rate of CLABSI, and for this we need proper surveillance for determining the frequency and then for the preventive measures. Majority of our incidences were attributable to our center, and major areas of emphasis include strict adherence to aseptic measures, that is adherence to insertion and maintenance bundles, especially maximum sterile barrier precautions, use of chlorhexidine preparations, and hand hygiene being the top of the list. Our conclusion is to reduce these rates, we are strictly trying to implement the insertion and prevention bundles, and we are promoting ultrasound-guided insertion for preventing multiple pricks, although we had quite a number of cases even where line was inserted using ultrasound guidance. And we are conducting regular surveillance using laboratory-confirmed cases. These are my references. Thank you.
Thank you very much, Dr. Safia Moin. And our third free paper is by Ms. Ayman Talha. She is a senior technologist at Khan Microbiology Laboratory. And her talk title is Gene Expert, MTBRIF Ultra Assay for Direct Detection of Mycobacterium tuberculosis complex in Suspected Extrapulmonary Tuberculosis Patients. Ms. Ayman Talha.
Thank you, Dr. Imran, for introducing me. Assalamu alaikum, everyone. I am Eman, and I am here to present Expert Ultra for the direct detection of Mycobacterium tuberculosis complex in suspected extrapulmonary TB patients. और नहीं है. There's no keyboard. So here's my declaration that I have no financial or any other affiliation with any commercial entity. It's almost like a breaking news that Pakistan has 1,100,000 new cases, and it's now 5th in the list of TB burden. Before 2000, in early 2000, it took around 2 months to detect TB and start ATT by using ZN staining and solid culture technology. But in 2011, GeneXpert significantly reduced the turnaround time from 2 months to 2 hours. Now, Ultra is revolutionizing the TB diagnosis with shortest turnaround time, that is less than half an hour, 1.5 hours. So we already have GeneXpert. Why do we need Expert Ultra? There is a comparison of their principles. In GeneXpert, we only have one that is semi-quantitated heavy nested PCR, but in Expert Ultra, with that, we also have high resolution MEL technology. In GeneXpert, it is single copy target type, whereas in Ultra, it has 2 different multi-copy target types. The reaction volume chamber, it is, I don't think it is visual before from backside, but the reaction volume chamber is almost large, twice as GeneXpert, and the limit of detection is almost equal to culture, that is less than 12 CFU per ml. Let's discuss the result interpretation. Expert and GeneXpert Ultra have almost same result interpretation, but Ultra is also detect trace. What is trace? Trace means that it has low, if we have low MTB in the sample, it could detect that. It is so robust, but the detection of rifampicin resistance or sensitivity is not that confident. So with Expert trace result, it is always rif resistant indeterminate. My objective of the study is to determine the sensitivity, specificity, positive and negative predictive value of the sample with suspected TB. It took me around 3 months to conclude my study. The design of the study is diagnostic accuracy. I take cases as extrapulmonary samples and controls as control as culture negative extrapulmonary samples. My sample is 32 CSF, 52 tissue samples, and 31 pus samples. In total, it is 115 samples. Out of 115 samples, that is, I have already told 32 CSF, 31 pus, and 52 tissues, 10 positive by both methods, 22 by Expert only, and 3 by mycobacterial culture only. The specificity, sensitivity, positive predictive value, and negative predictive value are written here. Let's discuss it in details here. The only limitation I face is that I have no prior information about antimicrobial tuberculosis if these patients are previously treated or not. CSF here is showing highest sensitivity, whereas pus is showing highest specificity. The negative predictive value is quite high, but positive predictive value, because of the limitation, is low. How does WHO recommending this? WHO recommend for the CSF, Ultra is the direct for the first line detection of TB meningitis. But in the cases of lymph node biopsies or in aspirates, it could be prescribed with culture and smear. Let's have a look on national data. There I have comprised 3 studies. I am sure there are many more. My findings are hide now, so no problem. Let's discuss the first study. Estesine et al. Performed a study of 1972 samples of extrapulmonary, that is CSF, pus, and tissue, giving 100% sensitivity and greater than 98.8% specificity with Expert. not with Gene Expert Ultra. I have done this study in Expert Ultra. And internationally, a 200 sample of studies conducted, which is showing 78% of specificity, which is closer to my study. Another study by Christopher et al., 52 samples, CSF pus tissue, exactly same I have done, and the same sensitivity and specificity. Here is a systematic review of 601 samples, and it is showing 100% specificity and sensitivity for the diagnosis of TB meningitis by using CSF as a sample. By the light of all these data and my study, I could easily say that Gene Expert Ultra is a capable tool for the diagnosis of extrapulmonary TB. However, MTB culture remains the gold standard because it's allows full range of DST on the strain. Here are my references, and I would like to thank Dr. Javedia and Dr. Raja to helping me in every thick and thin and allow me to help here and let me present this. Thank you so much.
Thank you, Ayman, for sharing the data with us. The fourth presentation is by Dr. Kuratul Ann Zahid. She is a resident at the Indus Hospital, and the title of her talk is Discordance between Gene Expert MTB-RIF and Conventional Culture Method for Diagnosis of Extrapulmonary Tuberculosis in smear-negative presumptive tuberculosis patients. Dr. Kuratul Ann.
Thank you for the introduction, Dr. Imran. Assalamu alaikum. My name is Dr. Kuratul Ann Zahid. I'm a resident microbiologist at Indus Hospital and Health Network, Karachi. And my supervisor is Dr. Nazia Khurshid. The title of my paper was Discordance between Gene Expert MTB-RIF and Conventional Culture Method for the Diagnosis of Extrapulmonary Tuberculosis in Smear-Negative Presumptive Tuberculosis Patients. Before I move on to my presentation, I would like to declare that I have no conflict of interest. Let's start with brief background and significance. Mycobacterium tuberculosis is one of the most ancient and deadliest bacteria, as we all know. It still remains a global threat despite advancements in the field of diagnostics, treatment, and infection prevention and control. It causes a communicable disease called tuberculosis, which primarily affects the lungs, but it can also affect organs and tissues other than lungs, such as it can disseminate to gastrointestinal system, genitourinary system, central nervous system, lymph node, bones, and joints. Tuberculosis is divided into two types according to its clinical presentation, which is pulmonary tuberculosis, in which cough and hemoptysis, weight loss are the chief complaints, and extrapulmonary tuberculosis, which can occur anywhere in the body other than lungs. Extrapulmonary tuberculosis manifests itself as a chronic granulomatous disease affecting body tissues and organs, as I said, other than lungs. Diagnosis of EPTB is a challenging endeavor for both physicians and pathologists, as it may not present with classic signs and symptoms of pulmonary tuberculosis, and also sometimes it is difficult to collect the specimen from deep-seated sites. There is variation in performance of Gene Expert MTB-RIF in detecting Mycobacterium tuberculosis in comparison with conventional culture in literature, depending on the geographical regions, which are low endemic regions and high endemic regions. According to the Global Tuberculosis Report 2021 by World Health Organization, Pakistan ranked 5th amongst the 30 high TB burden countries with estimated incidence of 510,000 cases, which accounts for 61% of the TB burden in the Eastern Mediterranean region. Pakistan also ranks 4th in the highest prevalence of multi-drug resistant TB. An upward trend is observed in extrapulmonary tuberculosis cases in Pakistan, which now accounts for 20% of the all incident TB cases. The main objective of our study was to identify discordance between Gene Expert MTB-RIF assay and gold standard bacteriological culture for the diagnosis of MTB and extrapulmonary tuberculosis specimens. Materials and methods, our study design was retrospective cross-sectional. Duration was from 1st January 2020 to 31st December 2021. Study setting was microbiology department of the Indus Hospital and Health Network, Karachi. Inclusion criteria, we included all the smear negative cases. Also, all the EPTB specimens which we chose for our study were the ones on which the AFB culture was performed on the same specimen as gene expert. In our exclusion criteria, we excluded all the pulmonary tuberculosis samples such as sputum, bronchoalveolar lavage, and tracheal aspirates. And all the AFB culture samples were excluded as well. After applying the inclusion and exclusion criteria, our sample size was 173. The results, gene expert results in culture positive samples, 69% of the culture positive samples showed that gene expert was detected in them, while 31% of the cases gene expert failed to detect MTB, while they were culture positive. Percentage of discordance, if we discuss it sample-wise, there were CSF, fluids, and gastric aspirate. 1% of the samples gene expert failed to identify, while in ascitic fluid and pus, there were 2% of the specimens in which gene expert failed to identify, and in tissue and pleural fluid, it was 12% each. If we discuss the number of discordant samples gender-wise, 25 of the specimens which gene expert failed to identify were from females and 28 were from males, while in 65 females it correctly identified MTB and in 55 males. Number of discordance, if we discuss it age-wise, according to the age groups, in pediatric age group there were around 54 culture positive cases in which gene expert didn't identify 15. And in adult group, there were 121 samples, AFB culture positive, in which gene expert did not identify 38. In our discussion, there are multiple studies which suggest that there is geographical variation in the sensitivity and specificity of gene expert depending on the regions. There is a difference in low endemic countries versus high endemic countries. Smear microscopy, although it is rapid and cost effective, but for it to be positive, there should be a high amount of bacilli, such as 5,000 to 10,000 bacilli per ml to be positive. Smear microscopy has high sensitivity but low specificity. Why? Because it can also detect NTM. Culture is still the gold standard and has high sensitivity, detects MTB when 10 viable bacilli per ml of specimen are available. But there is a drawback of culture that for detection it requires at least 4 to 8 weeks. While gene expert MTBRF has diagnostic accuracy, it is good enough to identify, to rule in the cases of MTB, but not rule out the cases. And I have discussed this, the geographical region variation. There is this study, this table is from a study that I have attached. In their study, you can see the upper arrow where their cases were around, I cannot see from here. Positive cases, I mean the LJ culture were positive in 28% of the cases, while gene expert was negative, and out of those, 82 were not detected in their study. Sorry, it's very small for me, I cannot see from here. You can see? Okay, thank you. So the conclusion of our study was that negative gene expert MTB results cannot rule out diagnosis of EPTB in endemic areas where there is high suspicion of tuberculosis. Clinical signs and symptoms, positive contact history, imaging studies, and histopathological diagnosis of chronic granulomatous lesion combined with positive microbiological culture will remain the mainstay for the diagnosis of TB. So our conclusion was that all the teams need to work together in order to diagnose these cases. It's not a one-man's job. Also, the limitations to our study were that it is a single center study. So our results may vary from other centers. The duration, in my opinion, 2 years is a very less time of duration to determine the results. And the sample size was less, 173. This is also my limitation. And I cannot generalize these findings onto other institutes. So these are my references. Thank you so much for listening to me attentively. Thank you.
Thank you, all the presenters. Is there any question for the presenter for free oral posters? Okay, Dr. Kausar, I have a question. Dr. Kausar.
I have a question from Purakulen. Why did you exclude the AP culture negative specimens?
Ma'am, because I was trying to see the discordance between the samples.
So there you can also detect.
I was only seeing the discordance in positive cases. I did not include negative cases. That was my main objective.
Khurat, could you explain what could be the possible reasons why? Because we think about that the molecular methods are better than the culture methods, and its sensitivity generally expert is high in comparison with the smear as well as culture. So what could be the possible things that can lead to this kind of discordant result?
Gene expert MTBF has a lower limit of detection of 114 to 131 colony forming units per ml. I'm talking about gene expert, not ultra. Our study was performed by gene expert. My study is on gene expert MTBF, not ultra. Ultra has a lower limit of detection of 16. So if the number of bacilli in sample are less, because for culture we need 10 viable bacilli, but for smear microscopy and gene expert to be positive, we need around 114 colony forming units. So it is very much possible the gene expert will be negative, while culture will be positive in those cases. And also we have to rule out a laboratory error and everything from our end before making conclusions.
Okay, best of luck all the presenters. Results will be announced tomorrow. I would like to thank all my esteemed presenters who presented and shared their expert thoughts. Now I would request Dr. General Irfan Mirza to please conclude the session. Thank you.
I think we had a wonderful session, pre and post lunch session. In the pre-lunch session, obviously, there were papers by the experts, by Sarah Griffith, then Professor Afia Zafar, and then Bushra Jamil, all related with the outbreak investigations, starting from Sarah's and then ultimately the infectious disease outbreak in the flood-affected areas. And then obviously after post lunch, we had a very fruitful and very nice lectures. All were very nice, and I congratulate all the audience, all the presenters for their valuable work, and I wish them all the best. It's very difficult. I think it will be very difficult for the assessors to choose which one is a better paper, but I think you people have all done a marvelous job in preparing your studies and presenting it. And the questions asked by the experts speaks of itself, the quality of work that you have put in. I think so. Video, yes, Dr. Saras. I forgot. I forgot. Obviously, I'm sorry for that. It was a very nicely, compiled, and I don't know, you did it yourself or you hired some professional to edit it, but it was very nice. It was very nice. But at the same time, dukh bhi hota hai, or but very, very nicely done, I think. So with these remarks, we have to now close, and obviously the viral shields, we have to give the shields to the presenters. So, नहीं है? बस ठीक है. तो समझे कि आपको shields मिल गई हैं. ये तो और चले जी. Thank you.
So we are about to start the next session. Please, we will be starting early so that we can reach in time. So may I request all of you to please keep sitting. Dr. Javaria is soon going to start the session.
Assalamu alaikum, everyone. Please, let's settle down and we'll start our next session. This is the one on, as you can see, this is the session on viral infections and the changing epidemiology and diagnostic challenges that we face, especially in this era of a new type of infection, you know, literally every year, or shall we say every quarter. So let's see. Today we have three speakers in our session. Our first speaker, and before we start the session, I'd like to invite Dr. Bushra Jamil and Dr. Asim Beg to chair and co-chair our session. I'm sure everyone knows Dr. Bushra and Dr. Asim. They've been, you know, Dr. Bushra is the current MMI DSP president. Dr. Asim has been treasurer for MMI DSP for so long, and both of them have worked tirelessly for our society, not just a society, but also for public health, which is a very important aspect that we seem to forget, that we don't just work in silos, we work for the nation. And that is something that this is the reason why we have both these champions right here as chair and co-chair, because we're looking at our emerging infections, and this is what we're going to talk about. So our first speaker is a very well-known Professor Dr. Imhan. She's chair pathology and lab medicine at Khan University. And of course, her huge amount of impactful work in arboviral diseases and the work on dengue, chikungunya, and of course, let's not forget SARS-CoV-2 that she's been working with our, the whole area, multidisciplinary work with the molecular and infectious disease and pathogenesis and everything. So let's invite Dr. Iram to the podium, and she will be talking about the changing epidemiology of arboviruses, dengue-like illness in Pakistan, and diagnostic challenges.
Thank you, Javeria, for that very explicit introduction. So, Assalamu alaikum, everyone, and a very warm welcome to all of you. What I'm going to talk today about is something that we all have been hearing a lot about this year and has been exciting all of us. Dengue-like illness. Everyone has been talking that besides dengue, perhaps there is something that is going on in Pakistan. And what are the evidences? What is it that we know? So my talk is primarily going to focus, well, so I have no conflict of interest, so quickly moving on. I have okay, let's go back. Okay, so what I'm going to talk about is a little bit outline of my talk so that you don't get lost when I'm talking, because I may talk a little faster. So global situation of acute febrile illness, where we are at this point in time, दुनिया भर में कहाँ जा रहे हैं, क्या नक्शा है, कौन सी illnesses spread हो रही हैं, current situation dengue की पाकिस्तान के अंदर क्या है, how dengue has evolved and how it is spreading, what are the other arboviruses that we are seeing in Pakistan as a part of a dengue-like illness, what is the local evidence, do we really have this evidence that there is dengue-like illness in Pakistan, or is it just a hoax that there is a dengue-like illness? And what are the challenges in diagnosis of arboviruses, and what are the way forward that we have? So we all know that acute febrile illness is one of the biggest global challenge throughout the world. And not much has been talked about in case of a non-malaria acute febrile illness, because whatever we talk about in the past was mostly linked to the malaria or the etiology-based illness. But when it comes to the undifferentiated fever, or fever where you do not know etiology, not much published data is available. In many of the studies, even if they are very well designed and high-powered diagnostics are used, the etiological agents remain unknown in 75% of the cases. So that is the major limitation that we have. And we all know that fever is the main cause for acute initiation of antibiotics, and therefore the burden of antibiotics also increases. And in healthcare providers, especially in LMICs, 32 to 74% patients, they end up taking antibiotics when they have fever. So in case of fever, this is what the world is overall looking at. You have malaria as a big entity, where they are symptomatic and non-symptomatic patients who present with malaria, and similarly you have asymptomatic, potentially dangerous pathogens who patients may present with them. And then potentially severe symptomatic incidents with the pathogenic etiologies like dengue, CCHF, leptospira, and other disease. However, this red circle over here is telling us a huge majority of the patients who present with non-severe, self-limiting, non-malaria acute inflammatory fever. So this is a big burden on public health throughout LMICs, including Pakistan. Sorry, it went off very far. However, malaria got controlled after the advent of this rapid detection technique, that is the ICT, and after ICT, the number of cases in malaria have gone significantly down. If you look at this graph, this is from the main endemic disease in the Africa. The red bar is primarily the malaria confirmed cases, and blue are the malaria non-confirmed or non-malaria infections. The yellow arrow is telling us when the, let me see. So this yellow arrow is telling us when RDT was introduced. As soon as the RDT got introduced, the number of malaria cases started going down, and the number of non-malaria causing acute inflammatory, acute febrile illnesses went up. And this is the same story all over in the endemic region. So RDT played a significant role in control of malaria, but what was happening before that, everything that was not malaria was not being diagnosed and was empirically being called as malaria and treated as malaria. Now that the diagnostic test is available, we know that there is a big chunk that is not malaria. So what is this non-malaria acute febrile illness? What are the etiologies of this non-malaria cases? So if you do the Google search, you will find a very little individual studies with large numbers. The studies are there, but the number of population that has been used is very small. However, this is the fantastic review that has just recently been published. And what they have done is that they have looked at the etiological agents in all the studies that have been published between 1980 to 2015. I think this is the most comprehensive study that has been published. And what important thing is that they have taken the two major areas where that is of our interest as well, that is the Asia and the Southeast Asia region. And they have included studies, not only the seroprevalence studies, but almost every kind of study that has been published. So these light blue are the case studies, the fever series, or the seroprevalence study. And my interest was because Pakistan, studies from Pakistan were also included here. And you can see largest number comes from India, and in here it comes from Malaysia. The region is very well represented in both the cases. So this is a very fantastic study if you want to study the causative agents. Now, what causative agents did they find out? If you look at the most commonly reported bacterial infections, in case of Asian countries or the Asian region, among the bacterial, of course, Salmonella typhi took the precedence, but in Southeast Asian region, it is the Burkholderia pseudomallei, which is taking the precedence. So these are, sorry, before I should also mention that the red dots are the mode of, these are the mode of transmissions, the aerosol, contact, and the food and waterborne, and the purple one is the vector-borne. Okay, so vector-borne bacterial infections are also increasing, especially the Susugamoshi and other rickettsial infections in both the regions are there. However, if you look at the viral etiology, again by the mode of transmission, dengue has taken the precedence, both in the Asian region as well as in the Southeast Asian region. It is the most at the top etiology of all the acute febrile illnesses that they have compiled so far, the studies that have been published so far. So I also looked at the prospective study that is being published, and this is the most recent one, which has been conducted in China. They did the Sentinel Surveillance Project in 2017 and 19 to see what are the acute non-malaria causing illnesses that are taking place. And they have used the most, top of the technology, TACMAN array card, and have included almost all the possible bacterial and viral causes that they could think of so that they could diagnose them in a timely manner and to come up. So what were their findings? The findings were again the similar. The dengue took the precedence. You can see the dengue over here. This is the most common etiological agents, followed by all other bacterial things, which have included primarily the Ornacea and Rickettsia, which were again most common that they found, along with other etiologies. However, the most interesting thing that I found was dengue, although was the most common that they found, but look at the type of the pathogens that were still not able to be detected. 57% and almost around 75% in two cohorts that they studied, they could not detect any pathogen, despite the fact that they were using microarray card, which are highly sensitive methods of using. So world is still struggling to find out what is the major causes of acute febrile illness in the region. So what is the situation in Pakistan? Where are we on this? So this is the data which is coming from the clinical laboratory of Aga Khan University, and you all know it is one of the largest university clinical lab where we have the largest data that we could present. So data log starts from more than 100 over here. The 0 is maximized to 100. And I've taken the top 4 infectious diagnostic tests that we perform, and look at the way that the numbers are increasing. So here, I've included dengue fever, all positive. These are all positive cases, malaria, typhoid, paratyphoid together, and acute viral hepatitis. What we have seen that hepatitis diagnostics also comes seasonally, increases just like dengue. But in both the cases, you can see malaria, no doubt, is still there and causes a significant amount of number of cases there, but dengue during the seasonal portion of the year, it takes up the precedence. Although it is present throughout the year, so we can say that it is now endemic, and you see the cases throughout the year. Same is the case in year 2022, and I can assure you this has been since past 5 years. We have been seeing all 4 top diseases, but dengue takes up the precedence, and the number has been huge this year. So what is dengue story in Pakistan? How dengue has a little bit about the dengue evolution in Pakistan, and then we will move on to the dengue-like illnesses. We have seen that dengue outbreaks are taking place not since 2006 we have reporting, but the precedence goes, or the history goes even beyond that. In our lab, we have also shown that it's not just one serotype, but there are multiple genotypes that are circulating in Pakistan, and I'm going to talk about it. And when you look at the genomic picture of these viruses, you will see that they have evolved in Pakistan in a very specific way. So what we have seen in the genomics is that in dengue 2, there are 2 major clusters that have evolved. The cluster that evolved from the southern region was quite distinct from that which evolved in the northern region, suggesting that there are perhaps point mutations that are helping this virus adapt in a different environment. And in Dr. Bushra's talk, you also heard that the temperature changes are also helping these viruses and vectors to adapt. Dengue 3 has also a very typical finding, which is the similar cosmopolitan type, which is circulating throughout the India and the region. These are the point mutations that we had noticed in type dengue 2 isolates, and these two are very typical to Pakistan, and this one is the very unique one, and this was seen in the northern region where the virus is adapting very well and the vector is adapting quite well. So this is the story that we have, and very recently NIH has published whole genome sequencing of the dengue 2 serotype from Pakistan. And here, although the number of isolates that they have used is very small, but they have matched it with the previous one that we reported, and it looks like the same cluster that is from the southern region is now expanding into the northern region as well. So the bottom line is that the virus is adapting very well in Pakistan, and it is going to stay here. Just to show you what the story of the dengue evolution has been, the reports of dengue type 1, and here are the legends for this. Dengue type 1 and type 2 have been reported in the literature since 1962, and these were the imported cases into UK from where they were reported. However, there are no just the case reports, no studies then. However, the first report that we reported from dengue was in 2006 and 5, where we found that dengue 2 took the precedence, and at the same time, dengue 3 was also identified. So there were a huge, hemorrhagic fever patients who were presenting because the population was already exposed to dengue 1 and 2, and then superimposed dengue 3 came in, so that more severe cases started coming in. Both dengue 2 and 3 then automatically, epidemiologically spread all over within the Sindh region, starting from Karachi to more into the upper Sindh. And our repeated, testing of the samples that we receive in this area, we consistently find type 2 and type 3 to be present there. It was in 2009 and 13 for the first time that the outbreak situation went worse in the KPK region and in the upper northern region, where type 2, and in addition to type 2, type 4 of serotype was also identified. So this was for the first time that the type 4 has been reported. During this time, we started the acute surveillance study that was in 2015 to 2019 throughout the Sindh region, starting from Karachi up to the Sakhar area, which is the upper part of the southern side of the Punjab. And what we found is that throughout the region, there are dengue type 2, type 3, and some samples along from this bordering region, we also found to have type 4 circulating here. So what is the story now? In 2022, you have heard Dr. Bushra saying that the dengue cases were coming, and severe dengue was also there, pre-flood region. And what we did the typing of this new samples coming from Karachi, and most of them, instead of type 2 and type 3, we are now seeing type 1 coming up again. So that is the reason why we are seeing more severe dengue in this time of the year. But this story is not just limited to Sindh region or to Karachi. This was a limited study that we did in our lab, but very recently NIH has reported out, and they have said that the spread of dengue type 1 is extending all the way from Sindh to the Punjab and KPK region. We are super endemic with dengue, so we have a hyper endemicity of dengue. All 4 serotypes of dengue are now circulating in Pakistan. So quickly moving on to this is the study that NIH has very recently reported, just came out last month, and you can see the blue is DEN 1, DEN 2 is orange, and DEN 3 is red. All 3 of them are circulating in Pakistan, in different cities of Pakistan. So to summarize, then 2, 3, and 4 that are circulating since 2004 and 11, they are likely to have been introduced via southern route of Pakistan. Both DEN 2 and 3 have undergone in situ evolution, and they have generated the heterogeneous population, which is now expanding. DEN 2 spread to northern area from to Punjab and KPK, and probably because of the mass, transmission of the, population moving in and out of both the regions. In 22, however, we are witnessing that there is an introduction of DEN 1. So this was the story of dengue. What about dengue-like illnesses in Pakistan? Do we really have other infections that are circulating? There have been news reports, a lot of news reports, but if you go and do the searches for if any particular study that has been published, we only get to see the case reports. And these are very recent case reports which are presenting like dengue, but most of the time, unfortunately, the testing has not been done completely, the complete investigation is not done, only NS1 antigen is done, which is incomplete investigation, and therefore labeling it as dengue negative is not correct. So this created a lot of international media attention as well. Telegraph also reported, and a lot of media coverage was given to this invasion. So what are the evidences? What's the evidence of dengue-like illness in Pakistan? So obviously there are no studies, but what I did is I've took out the data from our own clinical laboratory, where we have looked at the samples that came out negative for NS1 antigen, IgM for dengue, malaria, hepatitis A and E, and all the 4 top infectious diseases that I showed you that are requested. And to look at these numbers, they are progressively rising because they are negative for all of them for the one that they were. Even if you look at the dengue positivity, during the peak numbers, when they are highest number, positivity for dengue remains 55% to 52%, which again suggests that there is some etiology that is seasonal, that is coming along with dengue. However, we are not being able to diagnose at this stage. So I went back to look at what we had done in our 2015-2017. Our hypothesis at that time was also that not all patients that are presenting with dengue-like illness are suffering from dengue, and that other arboviruses are also circulating. So the focus here was only on arboviruses. Unfortunately, we did not look for any of the bacterial causes. Within the arboviruses, this was our trend. We could see that the numbers were peaking during the summer and the monsoon season, starting from the mid-April and all the way in August, September till October, and then peaking down. So was this everything due to dengue? No. Only 17.3%, which is around 170 patients that we turned out from 997, they tested positive for dengue, both by NS1 antigen and by PCR. And a small majority, say up to about 3.1, we were confirmed cases for chikungunya virus in this surveillance, but they were 77% of our patient population remained negative for dengue. So what were the other evidences? So what were the, sorry, let me just go back. I think I missed out one slide. Okay. Right. So what are the challenges? Why we were not able to, or why we are still not able to identify that mysterious non-dengue or dengue-like illness, but the causative agent for that? You see, there is a lot of at the clinical diagnosis level, there's a lot of overlap of the presentation of the disease itself. So even in our study, we saw that both the chikungunya as well as patients with dengue, they all presented with the same kind of presentation. Fever, of course, is the starting, but GI involvement, respiratory, neurological, hemorrhagic disease, they all were. But if you look at it closely, some of the differentiating points were there. For example, hemorrhagic disease and acute gastric involvement along with the fever was more commonly seen in dengue as compared to chikungunya virus. So we could delineate some, excuse me. So we could differentiate something based on close observation of the clinical presentation. However, when we looked at the ELISA-based testing, you can see that dengue virus, West Nile, and Zika, all 3 of them are flaviviruses, therefore they belong to the same group of viruses. A lot of cross-reactivity of antibody is seen, especially in the backdrop of dengue virus. If the dengue is circulating in the community, then it is nearly impossible for you to diagnose either West Nile or Zika based on your antibody testing alone. 74.5% of our testing for West Nile IgM also tested positive for for dengue. So it was literally impossible. However, at the same time, 48%, that is 7.4%, were those which did not cross-react with dengue, and they were purely West Nile positive. Similarly, 55, which is 8.7%, were just Zika positive. And these were further explored, and so just to give you the perspective, these cross-reactive and positives were seen all over, from Karachi, Hyderabad, Mirpur Khas, Sakhar, and Larkana. So it was not just restricted in Karachi, but antibodies were positive for all. So for this Zika virus cohort, what we did the PRNTs, which is the test of gold standard test where you really need to wear it out whether these pure antibodies are either due to Zika virus or due to dengue virus. So we did the PRNTs, and Kehkashan is here, and congratulates to her because she very well optimized them. So you can see that these, even at the dilutions like 1:10, 40, and 160, we could see that there was 50% reduction in plaque neutralization, and then those were confirmed as Zika virus. However, and similar case was for the West Nile virus. This we have already reported. Zika we are going to report very soon. So this, even for the West Nile virus, we found PRNT to be at 80, that is 80% reduction with PRNT testing. So both Zika virus, chikungunya virus, and West Nile virus, they are circulating. So these are the three most common probabilities of non-dengue-like illness in Pakistan if you can come across patients to see that. So what are the challenges for the PCR-based? One may ask, you also did PCR, what happened about the PCRs? The time is very important in cases of taking the PCRs. So there is often a very short viremic phase, average is about 3 to 5 days, and this is especially true for West Nile virus and JE virus and Zika viruses. Type of the samples tested. We tested only blood samples, but the more the sample type you have, the higher the yield is, which includes your CSF and urine samples. All samples should be tested together, not just one sample, but as many samples as that you may have. And then, of course, the sensitivity of the assay itself also varies. So depending upon the protocol that you are using, some protocols, they can test as low as 100 copies, but others may not. So we did do the PCR, and you can see, since dengue virus, viremia lasts longer, so you can pick them up. So majority of the dengue, 170 positive cases, all of them we were able to pick it up. Different genotypes, but we were not able to pick up any of the Zika or West Nile and JE virus. But same as like for chikungunya virus, the viremia persists for a longer time, up to about 10 days, so therefore we were able to pick up signals on PCR for chikungunya virus. So not everything is bleak. There are some advances now, so we can rely on many newer advances. One of the approach that we also talked about early this week is the syndromic approach. Multiplexing PCR techniques are available, and new cohorts, the cards for the specific tropical fever are now coming up. But the important thing is they should be driven towards the epidemiology of the region where you are practicing, so it is important to keep that in mind. Another important thing is besides using the diagnostic antigenic or DNA-based testing, and additional testing using immune markers are also coming up. So lots and lots of new immune markers have been linked now to these arboviruses, so we can capitalize on them. So using together both the antigen targets as well as the immune targets together would be the way forward in future. But last but not the least is the availability of metagenomics, which is, of course, is now the point of attraction. And a lot of papers are now being published where very unique pathogens are being identified, toxoplasma in one of the study, and in this particular study in Philippines, they were able to identify leptospira as one of the leading cause, which they could not identify earlier. So my conclusion is that comprehensive etiological knowledge about the non-malaria, non-dengue, acute febrile illness in Pakistan is still very scarce. Arboviruses and zoonotic bacterial infections such as brucellosis and Q fever are the most common etiologies that have been reported from Asia, so probably we should also start looking these in Pakistan as well. And most importantly, we need large multi-center studies. We need people from all centers. Not one center alone can give the answer of what is prevailing in Pakistan. So prevalence studies need to be done. Diagnostic tools, especially point of care, needs to be prioritized because that is how we started diagnosing malaria and dengue, and this is the way forward, including biomarker-based, I think, not only because to identifying West Nile in early stages would be good, but if you miss that early phase, then you may not be able to pick it up unless you have good biomarkers that last longer. So I think this is my last slide, and I would like to acknowledge all my collaborators who have worked with me, Dr. Kelly Barr and Dr. Maureen Long from the University of Florida, Dr. Javeria Faruqi, you all know. She's been instrumental in study design. And last but not the least, Kehkashan Imtiaz. A big hand to her because she's the one who's optimized most of the assays that we use. Thank you, everyone, for your attention. I think I did not. Did I go over time? Yeah, thank you.
Thank you so much, Dr. Aram. And there's so much to know, and there's so much more to explore, at least in Pakistan, yes. So our next speaker is Dr. Sinkins. He's a professor of microbiology and tropical medicine from University of Glasgow at the Virus Research Centre. And his research interests are Wolbachia, mosquito, arbovirus relationships, and their interactions, as well as the Anopheles epidemiology. And he's basically doing a lot of translational research on the control of mosquito-borne viruses, which is something that we need to talk about, considering the emergence of this group of infections, and at least in our region. So, Dr. Sinkins, I suppose you're online. Can you hear us?
Yes, hello.
Oh, we can't hear you. Just a second. Okay, I think you may be muted.
Hello,
Yeah,
can you hear me now?
Dr.
Can you hear me?
Just a second. Okay. You're not mute from our side. Can you hear us? Can you just wave and say yes?
Fine, yes. Can you hear me? I'm not muted on my side.
No, he can't hear us either.
I can hear you.
I think, Dr. Sinkins, can you hear us? Can you just raise it? Okay. Okay. So just a second. That means we can't hear you, and we need to sort this out. Would you like us to play your recorded video? Okay, and then we can give it a little bit of time and sort out the connection during the for the panel discussion. Wonderful. All right, thank you. So I do have your recorded video, and we will play that. My apologies, everyone. We've managed
The
to connect.
Wolbachia are maternally
There we
inherited
are.
or maternally transmitted intracellular bacteria. They're found very commonly in many insect species, including many mosquito species. And they're able to spread through insect populations by manipulating the reproduction of the host. The most common form is called cytoplasmic incompatibility, which is a pattern of reproductive sterility. In particular, when the male carries Wolbachia, shown here in blue, and the female does not, the offspring will not develop. There will be early embryonic arrest. However, when both male and female carry Wolbachia, there's rescue, and the larvae will develop, and at a high frequency will carry Wolbachia because of its maternal transmission. So this gives a reproductive advantage to the Wolbachia carrying hosts. It's a frequency dependent advantage. There is a threshold that needs to be exceeded because of effects like fitness costs at very low frequency. They will outweigh because of the infrequency of males encountering females that carry Wolbachia. They will outweigh the advantage if there is a fitness cost. Once that threshold frequency is exceeded, then Wolbachia will spread through, will invade that population. Now, it's also been discovered that some strains in some hosts of mosquitoes can inhibit viruses. In part, this may be a protective effect against pathogenic viruses in terms of its evolution. So when you combine those effects, the ability to spread through populations and to block the transmission of viruses such as dengue by mosquitoes, then we have a system that can be used for disease control. So I'll be showing you how we've gone about using this system in Malaysia in collaboration with the team at the Institute for Medical Research in Kuala Lumpur, IMR. So the primary dengue vector, Aedes aegypti, does not naturally carry Wolbachia. The secondary vector, Aedes albopictus, does carry Wolbachia. In fact, it carries two strains called W. albae and W. alb. So both of these species are very difficult to control with insecticides. However, when Wolbachia can be introduced into Aedes aegypti using embryo microinjection, we can transfer into Aedes aegypti, and what we get is a much wider tissue distribution of these Wolbachia. In Aedes albopictus, they reach quite high densities in the ovaries and the testes, the reproductive tissues. However, in somatic tissues that are important for virus replication, like the midgut and the salivary glands, those two strains are either absent or at low density in the native Aedes albopictus. Following transfer and selection of a stable Wolbachia-carrying line of Aedes aegypti, we can characterize the density in different tissues of various different strains, albae and alb from albopictus, and here in this example, we also have a couple of strains introduced from Drosophila. And we can see that they reach quite high densities in the salivary glands. And the same is true of midgut tissues, much higher density than in the native Aedes albopictus. This density does vary between strains. So one of the Drosophila strains, WAU, reaches the highest density. And there is a fitness cost, there is a reduction of lifespan, and that broadly corresponds to the intracellular density reached. So again, Aedes, in Aedes aegypti, WAU strain that reaches the highest density causes the most significant reduction in lifespan. And there is also a reduction in the hatch of quiesced or stored eggs, and that can be a significant fitness cost in some some conditions. And then when we compare these strains for their dengue transmission blocking ability, we see that, again, there's variation between strains. WAU, the high density strain, is the best in terms of blocking the transmission of dengue. And when you look at the dengue virus in the salivary glands, but WAU is also an efficient blocker, and it's also very stable when the larvae are raised at high temperatures with a diurnal peak around 36 degrees Celsius. The WLB strain maintains its density and maintains its dengue transmission blocking, in contrast to some of the Drosophila strains like WMEL from Drosophila melanogaster, which the dengue inhibition phenotype is much less efficient when the larvae are raised at high temperature because there's a fall off in density of that strain. So overall, the strain of Wolbachia with the best combination of characteristics in Aedes aegypti is WLB, the strain from Aedes albopictus. So we decided to take that strain forward for dengue control. The first step was introducing it to a local, recently collected background from Malaysia. It was created in an old Malaysian line, but we wanted to maximize fitness, competitiveness of the line, rather than a very lab-adapted line. And we did that relatively easily by backcrossing. And that process involves the Wolbachia-carrying females are mated with males that have been recently collected from the wild in the targeted areas, in this case in Kuala Lumpur. And then the offspring, again, the females are taken from the offspring and mated with the recently colonized males. And that process is repeated for several generations. So as I said, that will maximize fitness, which is really important. And it will also ensure that they have an insecticide resistance profile that is as close as possible to the wild population in order to avoid having a disadvantage associated with being more susceptible to insecticides. So for both of those reasons, it's really important to carry out this process of backcrossing. We selected various sites for our initial program of releases because Wolbachia is solely maternally transmitted. The only way to introduce it into populations is to release mosquitoes that carry Wolbachia. And so for this release program, the sites were selected, dengue transmission hotspot sites, and mosquito population monitoring was carried out using ovitraps to get an estimate of the population size so that we could scale the level of release accordingly. We want to minimize the level of release required. And then crucially, there was population, as well as population monitoring of the mosquitoes, there was also engagement with the local communities. Given that we have to release mosquitoes, including female mosquitoes that can bite, of course, then it's really important that communities are aware and are supportive. So these transmission hotspot sites, through a series of meetings, posters, social media, there was, in all cases, a very high level, a very high percentage of support, quite strong support for this program. One of the reasons for that is that dengue is causing big problems for these communities, and past efforts with various different approaches have not been very successful in reducing the incidence of dengue. So then a program of weekly releases was begun in these initial set of sites. And after a period of weeks, monitoring began using quantitative PCR of the captured mosquitoes, again using ovitraps for Wolbachia, so monitoring the frequency of Wolbachia using PCR. So this is an example of one of the sites with blocks of 17-story apartments, a site called Mentari Court. And on the bottom here, you see the results of the Wolbachia population monitoring over time with these weekly releases. In blue is the Wolbachia frequency, and in gray is the period in which the weekly releases took place, you can see that the Wolbachia frequency was going up pretty quickly. And then once releases had ceased, it remained at a high frequency with a small drop, but then returning to a high frequency. And in fact, now it's been several years since we did those releases, and it's still at a very high population frequency in that site. So then when we look at dengue incidence in this, in the Mentari Court site over a period beginning in 2014, and the releases began at the end of 2017. So you can see that the incidence is high. It's a transmission hotspot site, but with peaks and troughs, it's not consistently high. But once the releases start, then that incidence drops significantly. Whereas in the control sites, a number of control sites that were matched in terms of building type, we still see high incidence of dengue. So this pattern has been repeated consistently in our release sites. This is another example of 6-story apartment blocks in a site called Shah Alam. Again, in the 2 release sites, we see a reduction in dengue incidence compared to the control sites. In this case, the Wolbachia frequency rose, but then started to fluctuate and fall. So releases were resumed in that site, and then crucially in surrounding sites as well, in order to stabilize the Wolbachia in that population. So what was happening was there was immigration, quite high levels of immigration from surrounding sites of Aedes aegypti without Wolbachia, and that was making it difficult for the Wolbachia to remain stable in that site. So just increasing the footprint resulted in stabilization of the Wolbachia in that site. And again, it's still at a high frequency now. So there is a spatial element to the stability of introduced Wolbachia. So based on the consistent reductions in dengue incidence across our trial sites, it's now entered an operational phase in Malaysia. Ministry of Health are leading an expanded rollout of Wolbachia releases at an operational level in 31 sites in total now, with over 150,000 residents, all of which are dengue hotspot sites. And it's been very successful so far. There have been major reductions in the incidence of dengue across those sites. And crucially, if you have a stable Wolbachia in those sites, it can remain stable over a long period of time, and is thus a cost-effective control measure. It's not a continuous release program that's required. Once you've stabilized the infection in a site, it will remain at high frequency, as I showed you in the Mentari Court site, and no further intervention is required to achieve significant reductions in dengue incidence. So we can see over 90% reductions in some cases in dengue incidence. And the implication is, given that stability, is that we no longer need insecticide fogging, which is done on a case response basis and is relatively expensive. It's been estimated that the government of Malaysia, the health ministry, is spending over $100 million a year on dengue control with fogging. And so once you have a stable Wolbachia infection, you no longer need to do that fogging. And thus, those resources, both of the funds that were used for insecticides and the personnel, can be diverted into these programs for further rollout. And so another important factor is the fitness costs that we see between different populations. So we can see that here with different backgrounds of Aedes aegypti into which this strain WLB has been introduced. So we were doing that in preparation for expanding WLB Wolbachia dengue control program into other countries and characterizing the fitness costs. So this is really important because it determines how easy it will be to spread Wolbachia in different populations. If there is a very high fitness cost, a much larger scale of release will be required, which is obviously not desirable, and potentially a much longer period of release before you get the Wolbachia to be stable. So to maximize the effectiveness and efficiency of the program, we want the fitness cost to be as low as possible in those mosquitoes. So what's being shown here is in 3 different lines from Burkina Faso, Malaysia field line and Malaysia lab. We have the number of eggs produced when the eggs have been quiesced for a period of weeks, as shown here on the bottom, between 0 and 11 weeks. So dry stored, which can be important naturally for Aedes aegypti, particularly with ephemeral, temporary larval sites where the female mosquito will lay the eggs above the surface of the water, and then they may need to be, it may be a number of weeks or even months before they're immersed in water after rains again in temporary sites. And so this does vary. Sites with a lot of ephemeral larval sites would be much more affected by this phenotype than sites where there are more permanent sites like water storage containers. So what we see is that in the females that do emerge from the quiesced eggs, so there is a lower hatch of quiesced eggs, but in the females that do emerge, their ovaries can show a reduction in integrity, as you see here, compared to an ovary from a non-quiesced female. And we see that there are significant differences between the Burkina Faso line and the Malaysia lines in terms of the number of eggs produced, and crucially, there seems to have been adaptation in the Malaysia field line, so there is a greater number of eggs produced. So there's not a drop in the number of eggs produced even after 11 weeks of quiescence. And when we measure the antioxidant levels, there is a higher antioxidant level in that Malaysia field line. So again, there seems to be adaptation, which is allowing a lower fitness cost for these Wolbachia. So that is encouraging and will allow us to backcross and select to maximize fitness as we expand into different populations. So this antioxidant, higher antioxidant levels could be associated with the development of insecticide resistance that could be actually making it easier to spread Wolbachia through these populations. Okay, so I will briefly talk about what we've been doing to understand the mechanisms of how this works, of how viruses are inhibited in the mosquito by the presence of Wolbachia. And so we carried out proteomics to compare midguts from Aedes aegypti with different Wolbachia strains. And what we found was that there were perturbations in a number of pathways in these lines carrying the WMAL and WLB strains of Wolbachia in their midguts. There were perturbations in lipid transport and metabolism pathways that were very notable. And these lipids are really crucial for viral replication, for viruses like dengue. And using a fluorescently labeled cholesterol analog top floor, we see that there's sequestration in the lipid droplets inside the cells in the presence of Wolbachia. And when we release that sequestered or trapped cholesterol using cyclodextrins, which are shuttles of lipids and cholesterol with a hydrophobic interior, we see that there is rescue of virus replication, in this case of Zika virus, but we see the same for dengue virus after treatment with cyclodextrin. Now, for a different strain called WAU from Drosophila similans, we don't see the cholesterol sequestration phenotype, and we don't see rescue of viral replication after treatment of these cells with cyclodextrin. So what we do see using proteomics is upregulation of a number of RNA-binding proteins, at least 3 of which are known to be antiviral. So that's showing that there are different mechanisms between different Wolbachia strains. And so if we do see loss of efficacy of this strategy over time, for example, if there is evolution, if there is adaptation of dengue virus to restore replication in mosquitoes carrying the WLB Wolbachia, then we have the option of switching to a different strain, WAU, or any strains that we find that have mechanistic similarities. Because it's different to WLB, then that should restore the transmission blocking effect. The other possibility that one might see reduction of efficacy over time is if we see a reduction in Wolbachia density. So we have monitored mosquitoes from this population that I showed you, Mentari Court, which is one of the oldest populations that we have in Greater Kuala Lumpur, carrying Wolbachia. And even after 3 years in the field, that we didn't see any significant reduction in density of Wolbachia in the salivary glands. So that's very encouraging. That's probably something that will happen in the long term over time, that there will be a reduction in Wolbachia density, but we don't see that on the kind of time scale that would affect the efficacy of the control program, which is very good news. So this is my group in Glasgow, who have been behind the work that I've shown you, particularly Tom Ant and Marvi Mancini on the generation of Aedes aegypti lines. We also have a number of Aedes albopictus lines with different strains of Wolbachia that can block dengue transmission. And Steph Rainey and others on the mechanistic side that I showed you at the end of how Wolbachia can block virus replication. It's been an excellent collaboration with the team at the Institute for Medical Research, IMR, led by Dr. Nazni, and also, importantly, Professor Ari Hoffman at the University of Melbourne, who's been a very important collaborator in this effort in Malaysia as well. And funding has come from the Wellcome Trust. So as I said, the next stage now will be to expand these programs both in Malaysia and introduce them to other countries because we think it's a really effective, efficient and cost-effective system that can give long-term dengue control. I will finish there. Thank you. Hello, and thank you very much for the invitation to join you.
Dr. Sinkins here online, and thank you so much for this really amazing talk, and it has really opened our eyes regarding how we can use out of the box solutions. And you know, this relationship with Wolbachia is just baffling, but it's very useful. So we will be talking further. Yeah, we can do that after this talk, and yeah, we have a panel discussion. We will be we'll be taking your questions, so don't worry about it. And so our next speaker is, we have Dr. Kelly Barr. She is an associate professor at the College of Public Health at the University of South Florida, and her specialty is vector-borne and zoonotic diseases. And she's a dear collaborator. She's worked so wonderfully with us. We have, and her main areas of expertise are basically epidemiology and pathogenesis of arboviral diseases and zoonotic diseases. So we would like to invite Dr. Kelly Barr to please give us our talk. And hi, Kelly. I hope you can hear us. Okay, we can hear you.
Did you wish for me to share screen?
Okay, we can also hear you. We'll just give you the rights to share your screen, and then you can start your talk. After that, we'll be doing our panel discussion. Please stay on, and Dr. Sinkins will be there, Dr. Eram will be there, and we'll be taking questions from the audience as well.
I'm just waiting for the share screen.
Right, so Kelly, we're ready to go.
It still says host disable participant screen sharing.
Sorry, we're just about to give you rights. Just a second. Hi, Kelly. I think you're host now, so you can start sharing.
All right. Can you guys see that?
Yes, we can. Just need
Okay,
to go in presentation mode, that's all.
Great. All right.
Great.
So I'm going to talk to you about how the epidemiology of viral and vector-borne diseases is changing. So let's get to it. So when we're talking about vector-borne and zoonotic diseases in this day and age, we're talking about emerging infections. And when we're defining what is an emerging infection, that's when you have a new pathogen that's newly recognized, as we have with COVID-19. That's an emerging infection, but we also have infections that are known and are very well defined, but they're either increasing in prevalence in the areas where they're endemic, or they're emerging in new areas where they are not endemic. So Jones et al. Did a meta-analysis in 2008 of all emerging infectious disease events over a 60-year period. And it was really interesting to note that of all the emerging infectious disease, 60% of them were zoonotic diseases, and of those zoonoses, 72% were from wildlife. And here's a figure from this manuscript. This represents all the zoonoses that emerged from wildlife in the world. This represents the new zoonoses that emerged from livestock and domestic animals. And this represents the zoonoses and emerging infectious disease that emerged from vectors. So you can see there's certainly hotspots throughout the world. They seem to be more associated, especially the vector-borne, where we have Aedes aegypti prevalence. And so what these authors also noted was that the incidence of these emerging zoonoses and vector-borne diseases, they're significantly increasing. So when we have an emergence of a zoonosis, a spillover has to occur. So that's when the virus or pathogen moves from one species to another. And this can happen through just going out and petting wildlife. It can be through slaughter. Many times zoonoses, they're vector-borne because with wildlife, it's really hard to get the interface of human directly with the animal. So for a spillover to occur, they're very rare. Basically, all the stars have to align. There's several different factors. This figure from Nature Reviews really illustrates how difficult it can be. You have your reservoir distribution, their density, how prevalent the pathogen is in the reservoir population, the titer of the pathogen within the reservoir, can it be transmissible, how the pathogen's released, is it bloodborne, is it airborne, how quickly it spreads, how quick the incubation period is, and then the probability of human exposure. And then once you get to the human, we have our own immune system and our own barriers that prevent infection. So it's not easy. If transmission is going to occur and a zoonosis is going to spill over, it really requires close contact with the human and the reservoir. And the only way around this is with the vector-borne diseases. But then again, you have to have close contact of the vector with the reservoir and the human host. So spillovers, humans acquiring zoonoses from animals and domestic and wildlife is really increasing as time goes on. This is probably due to the increasing populations of humans are growing. I'll talk about it in a little bit. So when we have a spillover, that's a discrete event where an infection goes from a reservoir animal into a human. And from there, we oftentimes get outbreaks. So we are all familiar with vector-borne disease outbreaks. But when you have a spillover and then an outbreak, then we start getting human-to-human transmission. And we've seen this over and over again. Just recently with SARS-CoV-2, with MERS, with swine flu, with Ebola. And you can see, if you look at this figure, outbreaks are increasing. In the last 20 years, they've significantly increased. And it seems that the more travel we have, the more movement of people we have, the more commerce we have, increases the probability of outbreaks. And then this just shows, just talking about Ebola, which is a zoonotic disease, just within the past few years, it's significantly increased. So many times when we're talking about outbreaks, it's easy for the media to look at nations with limited resources, such as the Ebola outbreak in Africa, but that's not true. When we're talking about outbreaks and spillovers, they're more likely to occur in nations with a higher GDP. So we're looking at like North America, the United States, Europe, Japan, China, Korea. And so when we have a spillover, they can lead to epidemics, which we've seen many times just within the past 10 years. We're talking about high path avian influenza, MERS, Ebola, swine flu. And so zoonoses are a significant public health concern, and they are also significantly increasing. So this brings this question, why is there this increase in zoonotic infections? We're having new outbreaks show up everywhere in new areas where they've never been before. So the epidemiology we have known about these pathogens is we're having to rewrite it as we go, and it's challenging. And one of the reasons why we're having this increase is there's been a change in the way humans interact with the reservoirs. And it's mostly due to human-induced changes from human activities. So the major factor for this change in epidemiology of zoonoses is climate change. And so we're all familiar with how rising ocean temps has changed a lot of the climate patterns over the globe. We have desertification, and then we have these events of catastrophic flooding, which you guys just experienced there, where it displaces millions of people, and then it also alters the ecology of the vectors, because now we have water in places that were typically very dry. We have standing water for a much longer period that allows for vectors, but we also have the displacement of human and animals, and that changes the way humans interface with the animals. So when we're talking about rising temperature, I'm just talking about, this figure is showing the change in the incidence of Aedes albopictus and Aedes aegypti in the United States. And so we can see that both the range of Aedes albopictus and Aedes aegypti has significantly increased, and they're predicting it to significantly increase. So these individuals, they're going to be at risk for emerging infections like dengue virus, chikungunya virus, that they were not at risk of before, because the vectors being able to expand into new areas. And when we're talking about zoonosis, it's not just mosquitoes, all kinds of insects and vectors are expanding their ranges. This is just a figure of the southern United States and the spread of fire ants. Now, fire ants don't spread diseases, but they bite, they cause anaphylaxis. They're a major problem. They destroy crops. They were imported in 1939. You can see the small population here, and they stayed pretty static. The fire ants cannot handle frost. So that's what's really kept them contained within the southern United States. But here we have, with the rising temperatures, freezes are coming later. We're having winters in the Midwest where we may not even have a significant hard freeze. So now the fire ants are emerging all of the coast of the eastern and western side of the United States, and they're emerging into the Midwest. With the fire ant expansion, we've also have an expansion in the range of the 9-banded armadillo. So this, he's a rodent. They don't hear very well. They don't see very well. They're very slow moving. When they're found in the wild, they like to live in neighborhoods in suburban areas, and kids like to play with these and chase them around. Its range has significantly increased because of the rising temperatures. This guy doesn't have any hair. He's just skin, and so they're really limited to the warmer areas, but they've expanded up into the Midwest. And the problem with this is that the 9-banded armadillo is a natural reservoir for leprosy. So we're seeing in the United States an increase in the incidence of leprosy in children and hunters when they're out and about because they're interfacing with the armadillo. So again, we're having to rewrite the epidemiology. We're having to retrain physicians so they can recognize and treat these diseases, which they weren't trained to before because they're not supposed to be there. Desertification is another issue that's causing an increase in zoonoses. So desertification can happen two ways. One, we're all familiar with climate change. We're getting less rainfall, so the plants don't grow. But the other major way desertification is occurring is through the overgrazing of livestock. Here's a figure of just two pastures. This one's been grazed on, this one has not. Over here is some images from Turkey. And here we have sheep have overgrazed, and you can see when they have a high density, you have this sheep trace. And so this is just where residue from their urine and feces. And so this can alter the pH of the soil. They've eaten it, they've trampled it, and many times the native vegetation can't grow back. And they've noticed this in Turkey, that the vegetation quality has changed over the last several years. This didn't have a before and after picture, but they're seeing places where they used to have traditionally grazing lands for their livestock, but no longer have suitable vegetation. And with that, you've had herders and move their livestock, and so we're having increased density of livestock where there is a suitable amount of high quality vegetation. And with that, when we have the animals moving, they bring their vectors. So Turkey is noticing a significant increase in the spread or the incidence of Crimean Congo hemorrhagic fever virus. And that is due to climate change desertification. Another way that why we're having this increase in zoonoses is the way we're using land. Human population is increasing. We're building homes, we're building businesses. We have agriculture. We have to grow food to feed everybody. And here's just another picture from the United States. So this is the population of white-tailed deer. These are solitary animals. They like to live in the forests. They live in like suburban areas where we have houses with large yards. And over here, we have the population density and urbanization of the United States. And so we can see as urbanization has increased, so has the population of white-tailed deer. And this is not intentional. The United States Department of Fish and Wildlife issues a significant number of hunting permits. They have been trying really hard to keep the population down and away from people. They're not really associated with zoonoses yet. They do serve as an alternative reservoir for West Nile virus and some other vector-borne diseases that also infect humans. But they have not been associated with anything significant. So with the land use, and we have these large sprawling neighborhoods, these white-tailed deer have moved in. So they're living in people's backyards, they're living in causeways in areas where hunting cannot occur, and that has allowed their population to explode. And in the American culture, it's become really common for humans to feed the deer, to pet the deer, and so these wild animals they're really becoming domesticated. Many have lost their fear of humans, and so now we're having an increased interface of humans with deer. And this has led to the emergence of a new SARS-CoV-2 variant, just because we've had humans interfacing with deer. And so we're having, in real time, having to rewrite the epidemiology of this zoonoses, among others. And so it's really important as climate's changing and everything's changing, that what we know about zoonotic diseases and vector-borne diseases, what we've been taught in the past, may not be true now, because we're really living in a different world. So what's driving this emergence in new places is just human movement. It's crazy how much travel occurs. So we have shipping and transport. These are cargo ships. Red means more travel, blue means less travel, but we are all interconnected now. Look at this airborne travel, all of these flights. We could have people that have experienced a spillover or are infected with the zoonoses. Within 12 hours, you can travel pretty much across the world. And then something that's new and emerging is the movement of refugees. Now, in the past, refugees have been associated with war and conflict, but that is not the case anymore. We're having climate refugees, people being displaced by catastrophic weather events, or just the fact that they've lost their vegetation, the climate has changed, they can't grow their crops anymore, so they're having to move. And when we have this movement of like basic climate refugees, or just any refugees, many times they bring their animals with them. And so we have this interface. We have people living in tight quarters. We have animals and people being introduced to new areas. And so that really increases the probability of an emergence, but it also changes the epidemiology of these zoonoses because they're being moved into an area where they were never found before. A good example of this is chikungunya virus. So back before the year 2000, chikungunya virus was pretty much confined to Africa and Southeast Asia. And then because of cargo ships, Aedes albopictus and was introduced to North and South America, and infected mosquitoes were introduced other places, as well as travel of people. And so within just a short period, starting in like 2005 to 2016, that's a 10-year period, chikungunya virus emerged across the globe. And now it's endemic in areas, it's endemic in the New World over here, when it was just an Old World virus. And it's endemic now in Italy. And we have several outbreaks in Europe because the vector has emerged in Europe where it wasn't before. And so you can see these have to retrain physicians. They're not familiar with what chikungunya virus is. It presents most of the time like a febrile illness, but it can cause severe complications, neurological complications. It causes arthralgia, a chronic arthralgic syndrome, which can mimic rheumatoid arthritis. And so if we don't have our physicians trained to recognize this, people are going to be misdiagnosed. They're not going to be treated well. And also, if we're not recognizing the emergence of these zoonoses, they're going to go undetected, and that will allow for them to spread and infect more people and become endemic and lead to an outbreak or an epidemic. Another troubling emergence due to the movement of people is Rift Valley fever virus. So prior to 2000, Rift Valley fever was confined to Africa. It only circulated within the livestock there. And then shortly after 2000, it was like 2005 to 2010, we had a couple outbreaks, just small isolated outbreaks in Saudi Arabia and Yemen, and that was it. We have desert around here, and so it's really hard for vector-borne diseases to be transmitted where it's really hot and dry and the vector can't live very well, and also when you have a limited amount of livestock. But what we noticed within the last couple of years is Rift Valley fever virus. It emerged in the Kingdom of Jordan, which that's not too hard to believe because it's right up here next to Saudi Arabia where outbreaks had been identified. But what's really troubling is Rift Valley fever has been identified in livestock all the way up in Turkey and Eastern Europe. It's spreading into Iran and Iraq where it was not supposed to be before. So Rift Valley fever virus is a significant problem. Yes, it causes human disease. It causes blindness. It can cause abortion. It causes a febrile illness, but also it destroys livestock. It causes spontaneous abortion in goats and sheep and camels, and it can really destroy the livestock industry in an area. And so this is a real problem if we have an unknown zoonoses emerging where there are vector present and there are livestock and reservoirs present to maintain that transmission cycle. So with that, I'd like to thank you. We're still defining the epidemiology of these zoonoses because they are changing in real time, but it's really important that we're aware of the different presentations of zoonoses, and we keep aware with what's being reported in the ProMed emails so we can share with healthcare providers so they can keep an eye out, and we can share with animal workers so they can keep an eye out because we're going to need vigilance, we're going to need surveillance, and we're going to need cooperation and really a One Health approach to respond to these zoonoses. Thank you.
Thank you so much, Kelly. I mean, honestly, we are just, at this point, we're just, what shall we say? We're flabbergasted at the extent of this, okay? And nobody had thought, probably, that it would have been that big. But we're ready for our panel discussion now. So Dr. Iram and Dr. Senkis is also online. And Dr. Barr is also there, and we're ready to take questions from the audience first, and then we'll go on to a couple of questions or specific topics of discussion. Yeah, okay, Dr. Junaid.
Yeah. This is Junaid Iqbal. I'm an infectious disease researcher. I have two questions, one with Dr. Iram. Dr. Iram, did you also able to sequence this Zika virus strain that you guys have isolated? If yes, I've heard like there are differences between the strains that in the northern hemisphere and southern hemisphere, and one of them is more related to microencephaly.
So we are still in the process. We don't have the result yet, yes, but we have sent the isolate for this the whole genome sequencing. PCRs have turned positive, but we wanted to be absolute sure. So we did 2 different PCRs. Both were positive for Zika, which gives us a very strong level of confidence that that is a true positive, but yet we wanted to do this. So the answer is yes, we have sent this isolate for the sequencing, but we don't have the results as yet. So as soon as the confirmatory results come in, we will be publishing it soon.
Thank you. And the other question, if you allow.
Yes, sure.
Yeah, it's with Dr. Steve. I want to know about the environmental spillover of this Wolbachia strain from mosquitoes to other arthropods or arachnids, or from mosquitoes to other water bodies like rain puddles and others. Like, have somebody did the environmental spillover of this strain from mosquito to other bugs or like environmental samples?
Dr. Sinkus.
Thank you. Can you hear me? Can you hear me at all?
Just a second. I think we still can't hear you. Can you check on your end? Because
I'm not
we could
muted.
Get Kelly. We could hear Kelly.
Yes, I can hear you, Stephen.
And I think Dr. Sinkus can also hear me, but...
Yes, I can hear fine.
Oh, wonderful. We can hear you too.
Great, okay. Thanks for the question. So Wolbachia is only transmitted vertically from the female insect, in this case mosquito, to the offspring. It can't survive outside of the cells of the host, so it can't survive out in the environment. So the opportunities to be transferred in the environment to other species are extremely limited. It can happen very rarely on a very long evolutionary timescale, but it doesn't happen on a day-to-day basis. So it's a very clean approach. There is essentially no environmental contamination of other species. And actually the strain that we are using in Malaysia, WLB, is already present in Aedes albopictus, so it's already present in field populations of mosquitoes, just of another species.
Thank you so much. I mean, that definitely sorts out. Dr. Rumina has a question, and then I'm going to ask Dr. Asim, and then Ali. All right.
Thank you, Dr. Simkin and Kelly, for excellent and actually remarkable talks. My question to Dr. Simkin, when you in that area, you were following dengue. What was the impact of the release of Wolbachia-infected mosquitoes on malaria and on West Nile, if it was endemic in that area?
So they're not malaria vectors that we're targeting, so we don't have any effect on malaria. In fact, there isn't any malaria circulating in those areas. I think the same would be true for West Nile, but it's not something that is being detected in these populations. Now, Wolbachia does have a broad antiviral effect. So, for example, it blocks the transmission of chikungunya and Zika viruses, and there is a little bit of chikungunya circulating. So it would have an effect on the transmission and incidence of chikungunya in the areas where we have done releases. But it's much more difficult to detect because there's much less of that virus compared to dengue.
Hi, Stephen. This is Asim Beg from AKU. It's a follow-up question from what Dr. Rumina just asked. There was a study in Africa on Anopheles gambiae using Microsporidia MB strain rather than Wolbachia. Wolbachia mostly for dengue. I was just wondering, from this part of the world, we've had massive flooding, and the reports coming out from the government side that the strains of mosquitoes have suddenly, changed, and Culeus facies actually had less sporozoid counts for malaria, and what was reported earlier in literature, Stephensi is the main urban vector. Are there any natural things that we could actually use the same technology that you've used in Malaysia for looking at stuff like this here in Pakistan?
If you are asking specifically about malaria vectors, is that the case?
Yeah.
Yeah, so I think the Microsporidia work, that's That was a paper that we put out from my group in collaboration with Jeremy Heron in Kenya. And we're actively working on that, on Microsporidia MB. I think it would certainly be worth characterizing, doing some surveys of local malaria vectors. We've been concentrating just on Africa, but it's not restricted to Anopheles gambiae. It's also present, for example, in Anopheles funestus. So it would certainly be worth doing some surveys. It's a little bit different to Wolbachia because it doesn't manipulate reproduction of the mosquito, so it doesn't have a self-spreading system. But on the other hand, it does have a spore stage and it can be transmitted from males. So in theory, you could be releasing males and increase the prevalence of the Microsporidia, and it's a very good transmission blocker for malaria. Okay. So yes, I think there are great possibilities there, and it's a system that we're excited about and actively working on. There is also a low prevalence, low density Wolbachia in Anopheles gambiae in West Africa. So there are possibilities for using Wolbachia as well, but it's at the moment we're actively looking at identifying strains that could be used in Anopheles as well. That's proving much more difficult than it is for Aedes aegypti.
Okay, thanks.
Thanks for the question.
I'm going to ask Ali to take for the next question, and then Dr. Bushra.
Thank you. So my question is to Dr. Simpkins as well, and the question basically is, do we have any prediction models that have analyzed what will the effects be 5, 10, 20 years down the line on the ecosystem generally of introducing mosquitoes that are whose viromes or whose microbiomes have been altered? Is this a concern?
Well, the approach that we're taking is, as you say, is replacement. So the mosquitoes are still there, the Aedes aegypti is still there. So it would actually have very limited ecological effects. The main ecological impact would be the cessation of insecticide fogging, which increases insect biodiversity, and thus the predators like the birds of insects, the biodiversity and abundance will increase. But in terms of actual impacts, ecological impacts, as I mentioned previously, the Wolbachia itself aren't being transferred between species, and they're not. We're not suppressing those mosquito populations. We're replacing them, or we're not eliminating them. So ecological impacts, negative ecological impacts, we really don't predict any.
Okay, Dr. Bushra, would you like to ask a question?
Thank you very much. I'm Dr. Bushra Jamil, and I have questions for all three speakers who are here. Excellent presentations, all of them. So my first question is to Dr. Iram. You showed us a slide showing that the numbers of malarial fever go down with increasing use of rapid diagnostic tests. So do you have any similar data from Pakistan showing utilization of rapid diagnostic tests and its implications for malaria cases?
So not really a big study, but a small study has been published where people have shown the impact of the malaria in their hospitals after using RDTs and how they have responded, but not a big study. We haven't done any population-based study where we can very comfortably say that the malaria is going down in this part of the world because of the RDT availability. It has to be in the community and not in the hospital. So that's where RDT exactly. It has to be used there.
Okay. So my next question is to Kelly. Outstanding presentation. So in fact, I have two questions. We had an outbreak of chikungunya in Pakistan, and that was in 2016. And since then, we've seen very few cases of chikungunya. So would you like to tell us what actually dictates the rise and fall of chikungunya? Keeping the environmental conditions or considering them to be static, or in fact, they're more favorable for mosquito breeding, number one. And second question to you is regarding Rift Valley fever. So how is it that Pakistan has never reported any study or case of Rift Valley neither in animals nor humans? Does it have to be with lack of suspicion and diagnostics not being carried out, or are we in some way protected from Rift Valley, which I find it difficult to believe? So please, your comments.
Okay, so first I'll talk about chikungunya. So yes, you guys had a huge outbreak in 2016. And so with these vector-borne diseases, you'll have outbreaks where every, around 7 to 10 years in endemic areas, you'll have an outbreak. And so the same thing happened with Zika. It came to South America, it was everywhere, and then we really haven't seen it since. And so what happens is during these outbreaks, yes, we have lots of cases, but the cases you're seeing in the hospital represent maybe 1 to 10% of infections. So you have, out of every 100 people, you've got 80 to 90 people who have no symptoms but were infected and they've seroconverted. So what's basically happened is your population they're all immune. So we have to wait for those antibodies to wane. We have to wait for new people to enter that population. So we have a naive population again. And so you can either there's usually low level subclinical transmission, usually in the foresty areas. But because there's so many immune people, you're not going to have an outbreak and spread. So that's likely what's happening here. We experienced that as well with the chikungunya outbreak in South America. We had that several years ago, and then it's been 7 years and we're seeing another large outbreak again. So I would say within a couple years, you guys will probably be experiencing another large chikungunya outbreak. So that's definitely something to keep on your radar and to not stop doing diagnosis for it, because it will come back. So for the Rift Valley fever, now I've always felt that where you have travel and commerce between endemic areas and you have similar livestock as endemic areas, you should be doing surveillance. It's why you guys haven't documented is probably because you're not looking. It seems that wherever people start looking for Rift Valley fever virus antibodies, they're finding it. Now, maybe you have vectors that don't transmit as well. Maybe you have other zoonoses there that will cause abortion in your goats and your sheep. And so it's being misdiagnosed as something else. Lots of times when Rift has emerged in areas where it's not supposed to be, when people present to healthcare with the uveitis, they're diagnosed as having something else. Nobody even looks for Rift because it's not on the radar. So that's what I think could be happening with you guys. If they're finding it in Iran and Iraq, they're probably, I would be surprised if you guys didn't have Rift just with that highway that was built on, that superhighway that was built to transport goods from Africa through to China. And so if you're having import, you're going to easily transmit vectors if you're using, vehicles like trucks. It's easy to transmit vectors that way. I don't know if livestock is being moved, but I certainly think it's something you should look for, especially if you're noticing like abortions in your livestock, or you're having an increase in uveitis in people, or having an increase in abortions in women. That would be something to look for as well.
Especially when a large proportion of our patients have tested negative for everything which can be diagnosed here. So it doesn't mean that they don't have infection. Probably they are infected with something which we are not testing for.
Yeah, that's exactly what we feel. This, this gold mine has not been mined.
Yes, yes. And my last question to Dr. Simpkins. Regarding the cost of this Wolbachia strategy. So Pakistan has very limited resources, and there are many groups who tried to approach either Singapore or Malaysia to get this technology transfer. But I don't think any of the groups have been successful. And what some of the groups, at least one group has done, is focus on the cheapest possible strategy, that is Bacillus thuringiensis larvicidal formulation. And so what are your thoughts on this? Should we go for the cheapest possible, stick to it, or should we keep exploring and try to get the Wolbachia strategy as well?
Thanks. Yes. Well, it's certainly something I'd very much encourage to bring the Wolbachia approach replacement into Pakistan, and I would be very happy to try to support that in any way that I can, and certainly the use of the strain that we've used in Malaysia. So it is a cost-effective strategy, certainly compared to the alternative approach that's being used in Singapore, which is releasing just males for the sterility effect to suppress the population. That requires a much larger scale of release and is much more expensive. You also have to separate the males from the females. Replacement is just a discrete, limited period of releases required, and then you'll get long-term protection. If it's been done on a wide enough area and is stable, you'll get long-term protection. So it can be very cost-effective, and it can make use of staff that are already engaged in dengue control programs, can be redirected into this kind of program. The main expense really is the monitoring, which currently qPCR is relatively expensive, but we are working on lower cost systems actively, like LAMP. So hopefully within a short period of time, it'll be much cheaper to do that Wolbachia monitoring. As for BTI, it's certainly compatible with the Wolbachia approach, and I'd very much encourage continuing with BTI. There's no reason not to be exploring multiple approaches at the same time, because they're certainly compatible with each other, to run alongside each other, with the exception really that insecticide fogging is not a good idea to do in a Wolbachia invaded population, but we do continue to do BTI larval treatments.
So wonderful.
Last question.
Yeah, so Dr. Eram has one last question, and we'll take that.
So, Dr. Stephen, how did you manage the, did you see any resistance from the government, as these are considered as, genetic manipulation and releasing these genetically mutated, or they are like GMOs kind of things that they are considered. So did you see any resistance? Did your team, the Malaysian team in Malaysia, did they feel any resistance from the government? And if yes, how did they tackle that?
No, we had a very mutually supportive interactions with, IMR, the Institute for Medical Research, is the research institute of the Ministry of Health, so they're already close interactions. So they were very supportive throughout. I think it's important to note that these released Wolbachia have not been genetically modified. They have been introduced into Aedes aegypti, but they haven't actually been genetically modified. So we don't, in terms of the regulatory framework, we don't have to register them as a GMO. It's a form of biocontrol. Although they are vertically transmitted, they haven't been genetically modified. So that makes life much easier. And we don't get there were some genetically modified mosquito trials that were carried out previously in Malaysia, which attracted protest groups, anti-GMO groups. That was mosquitoes developed by the company Oxitec. But we fortunately haven't had to deal with those issues because it's biocontrol rather than GMOs.
Okay. Thank you.
Right, so I have one last question. So my basic question is, we're doing a lot of research, and in this time, especially with during COVID, we realized that we can do research in real time and then translate it, right? But I was just thinking for how can low middle income countries actually, take full part in it without, driven by the staff of WHO that you have to do this, because with us, probably the reason why we had that great response for COVID was because it was driven by WHO. It was a global pandemic. We had to do it, and we came up with solutions for it. So when we're not really driven, and yet we have the problem, apparently we can do translational research, right? It's been proven. But how can we motivate our LMICs to bring about that research to clinical practice, to public health immediately in real time? Any ideas? You've, Dr. Simpkins, you've been working with the government in the Far East, and I know Kelly has been working with us, but we are a private institute. I don't know how whether you've been working in South America with the government as well. What are your thoughts on that?
Is this a question for me, sorry?
For both of you.
Yeah. Well, I think from my point of view, from the collaborative group, it's really important to have local leadership of a program like this. I think when it's imposed externally, for example, there have been programs where the mosquito rearing has all been done out of country, and there hasn't really been local capacity building and training. I think that's a much less successful approach than when it's locally led. I think that's really crucial, really important. So I don't see any, as long as there's a baseline of entomological and virological expertise available, this is a kind of approach that can be done in any lower middle income or lower income country.
Yes, I agree. I think it's great for local universities and local institutions to take leadership and take charge of addressing this, because you're training the individuals who will go into work with these government organizations. Yes, it's important to have government buy-in and collaboration with this, but to rely on a Ministry of Health to oversee the entire country disease epidemiology control, that's not economical, that's not going to work. It needs to have an interface between local institutions, academia, and the government facilities, so you can work together, share what you have, share your training, and then have that infrastructure in place, so when you have an outbreak of something, or if you have an idea for implementing mosquito control or vector control or human healthcare policy changes, that it's easier to implement because you've all been working together. But I think it's important for local institutes to take ownership and just start contributing and helping with what doesn't matter how little it is. It doesn't cost a lot of money to look for mosquitoes. It doesn't cost a lot of money to go just gather epidemiological data from patients that are showing up to healthcare. It doesn't cost really any money to reach out to healthcare providers to make them aware of what's circulating in the region and what to look out for, and to provide training as to better triaging, recognition of symptoms, and infection control. So yeah, I digress.
So we all take responsibility. Good. Right. So we'll thank you so much for being here, Dr. Simpkins, Dr. Barr, Dr. Iram Khan. We're really, really honored that you have been here and you've talked to us. And we'd now like to move on to the next section, which is a free paper presentation by three papers that we have chosen. The first one is Dr. Rabia Sajjad, and she will be presenting a paper on the diagnostic accuracy of VIGEN-Q fever panel multiplex PCR kit in the rapid diagnosis of typhoid, malaria, and dengue. So, Dr. Rabia, please come to. Dr. Simpkins and Dr. Barr, if you'd like to stay, you're most welcome to. In fact, we'd love it. You know, you're also, we're not forcing you to, just that.
Bismillah ar-Rahman ar-Rahim. Assalamu alaikum. I am Dr. Rabia Sajjad from AFIP, and today I'll discuss this study, Diagnostic Accuracy of VIGEN-Q Fever Panel Multiplex PCR for the Rapid Diagnosis of Typhoid, Malaria, and Dengue. So I have no financial benefits or conflicts of interest to declare. So when we talk about diseases like malaria and dengue spread through the mosquito bites, and the fecal-orally transmitted diseases like typhoid fevers, so Pakistan has very favorable conditions for all these sort of diseases. And that's why when we talk of acute febrile illness in our setup, so these three are the topmost differentials. According to WHO, Pakistan is endemic for malaria, and in the year 2022, from January to August, 3.4 million suspected cases of malaria were reported in Pakistan. And about dengue, in 2022, from January to September, around 26,000 confirmed cases were diagnosed. And 74% of these were only in the month of September, which is probably due to those unprecedented flooding in our country that began after June, mid-June. And regarding typhoid, we, along with India and Bangladesh, are again in the most endemic region, and we are also fighting exterior Salmonella typhi. So, the importance of rapid and accurate diagnosis it cannot be undermined. Because accurate and rapid diagnosis would, of course, clarify the clinician's mind as he'll go for a targeted therapy. And also, it will cut short on the misuse of antibiotics, as malaria and dengue don't need antibiotics. So it might help in antimicrobial stewardship as well, which is a topic so close to the hearts of all of us, being microbiologists and ID physicians. So this study was planned with an objective to determine the diagnostic accuracy of a novel commercial multiplex PCR fever panel kit in the rapid diagnosis of typhoid, malaria, and dengue. So it was conducted at the Department of Microbiology, Armed Forces Institute of Pathology. It was a descriptive prospective study from July to September 2022. 60 samples selected by non-convenient convenient sampling were chosen. And all patients presenting at AFIP reception with a history of fever and who were prescribed any one or more of the tests for malaria, dengue, or typhoid were included in the study. And those patients who were not willing to give blood culture samples, they were excluded. So we took their samples in plain bottle EDTA, and of course we took blood culture samples as well with informed consent. So for malaria, we did the routine procedure, that is a thick and thin films. For dengue, we performed ELISA for NS1 antigen. And for blood cultures, typhoid was diagnosed on blood cultures. And similarly, at the same time, we performed this multiplex PCR testing. This is a real-time quantitative multiplex PCR that detects all these three, malaria, dengue, and typhoid. Data was analyzed by SPSS 24, and here are the results. So out of 60 samples that we tested, by the routine methods, that is the thick and thin film, and the blood culture, and the dengue NS1 antigen, 11 were reported to be positive. And by the multiplex PCR, it detected 12 as positive. And the breakdown of these cases, 3 cases were detected by thick and thin film, positive for malaria, 4 were typhoid cases detected on blood culture, and 4 dengue cases were detected by the NS1 antigen ELISA. Whereas the multiplex PCR, it correctly identified all those 11 cases, and in addition, it also was positive for another dengue case. And when we clinically correlated, the patient had clinical signs and symptoms of dengue, he had low platelets, but the NS1 antigen failed to detect it. So we made 2 by 2 tables for all the 3 diseases separately and then combined for the sensitivity, specificity, positive, negative predictive values, and the accuracy. For malaria, all the parameters were 100%, and so was for typhoid, whereas for dengue, the accuracy was about 98%. And the overall efficiency of the multiplex fever panel came out to be 98.33%. Now coming to the cost concerns. We calculated the cost of one test to be less than 2,000 rupees, so it is cost effective as well in that regard. Here is a little literature view. In a study published in 2019 in the Malaria Journal, it showed that microscopy and rapid diagnostic tests missed 12 to 13% of malaria cases, which were detected by PCR. And another one in 2019, published in the International Journal of Infectious Diseases, showed that dengue virus was detected in about 25% of samples by PCR, 19% samples by NS1 antigen ELISA, and around 10.5 samples by IgM ELISA, which shows the clear-cut superiority of PCR over these conventional tests. And regarding salmonella, a study was conducted in Bangladesh, and it showed that among 200 blood samples, 57 were positive for Salmonella typhi on PCR, whereas on culture, only 16 samples were positive. So again, there is clear-cut superiority of the PCR upon these conventional methods. We could not find any study that showed multiplexing of these 3 diseases, probably because this is a problem in our part of the world, so study should also come from our side. The BioFire Global Fever Panel, it detects 4 diseases, that is malaria, dengue, chikungunya, and leptospira, but not typhoid. So to conclude, this multiplex PCR is a very useful test addition. It is cost effective and consumes a lesser time, especially when we compare it with blood culture that is so time consuming. But the biggest limitation of this study, that the sample size was very low, and therefore it needs much more studies to validate it. Here are some of the references. Thank you all for your patient listening.
Thank you so much, Dr. Sajjan. And I find this to be quite heartening. Let's hope we get better options soon. So our next speaker is Dr. Najia Ghachi, and she's from Arkan University. She's associate professor, and her topic is pathogens identified by minimally invasive tissue sampling in Pakistan from preterm neonatal deaths, the PAP study.
Thank you, Tavia. So, can you please? So, I'll be talking about this study, which is conducted in EKU in collaboration with Department of CHS, where we are looking at the causes of neonatal deaths and causes of infection as a cause of neonatal death in those cases. So as you all know, that in Pakistan there are 40 deaths per 1,000 live births in Pakistan compared to the 17 across the world. New deaths are attributed to preterm birth, low birth weight, infection, birth asphyxia, and congenital anomalies. Infection is considered as one of the primary cause for more than 1/3 of the neonatal deaths, with the bacterial infections are the leading ones. So infections in preterm is higher compared to the term neonatal deaths. The common pathogens which were identified in various studies outside Pakistan reported GBS, ureaplasma, E. coli, and Staph aureus were the main causes. And when it comes to the viruses, there were cytomegalovirus, RSV, and parvovirus. There's a recent study from Africa reports 70% of childhood deaths at hospital-acquired infections due to Acinetobacter or Klebsiella and S. aureus. So the objective of the study was to assess the knowledge gap about the infection-related mortality in Pakistan. And this is, again, a sub-study of a prospective study already ongoing. And it was a project to understand the research in preterm pregnancy outcomes in stillbirths in South Asia called PURPOSE. The study was conducted at tertiary care hospital in Karachi. It was simultaneously conducted in India as well, but I'm presenting data from Pakistan. So overall, 1,400 preterm live births were recruited, and they were followed up to 28 days of life. 47 of them were died, 475 were died, of which 145 consented to provide placenta, blood, and tissue samples for the study. We did, all those samples were processed for PCR testing. The samples were stored in a preservative for the later testing. We did the total nucleic acid extraction using a QIAGEN protocol. We did the homogenization of the tissue samples to extract the total nucleic acid from the tissues. We used TechMin array cards, which can screen more than 54 pathogens in a single PCR run. The assays were run on QuantStudio 7 Flex real-time PCR system. The proficiency testing for these assays were done via CDC and CHAMPS protocol, which already run these kind of study across the globe. While we were doing any assays, we made sure that we incorporate all the positive controls, internal controls, and negative controls in the study, and the QC results were continuously monitored. So if you look at the demographic characteristic of maternal and neonatal population of the population included in the study, the majority of the females were the mothers were between the age of 20 to 30 years. They were educated for between 5 to 12 years of education. Mostly were homemakers, since this was a public health hospital, so public sector hospital, so probably that women who are homemakers and they do not work, they tend to go to public sector hospital because they are dependent for the healthcare to their husbands and the families. The good thing is the majority of them received antenatal care, and then almost 70% of our population was found to be anemic. Overall, the neonates, the babies born, were between the weight of less than between the weight of 1 to 2.4 kgs in birth. So if you look at the pathogens identified in the tested tissues, we have identified that approximately 21% of the tissue and 25% of the blood sample tested at the time of that was positive for Acinetobacter. There were almost 13% positivity among Klebsiella, and then we saw Haemophilus, some serotypes of Haemophilus influenza. Other pathogens were not identified. Interestingly, no ureaplasma or GBS was identified. We further looked at the day-wise breakup of death among the children, and between day 0 to 28, we saw 24% Acineto as a cause of was identified between day 0 to 28 in those cases, and then Klebsiella was 14.6%. We further looked at the day-wise breakdown of the deaths among those babies, and what we have noted that after the day 4, there's an increase in the positivity of the Acinetobacter, and we were seeing more Acinetobacter as the children are aging. That is showing that there is a nosocomial transmission of infection happening and leading to the infection in the babies who are admitted in NICUs. Majority of these babies were admitted in NICUs. So we also looked at the placentas of those babies, and we screened for all those pathogens in those placenta tissues. And the pathogen which was identified was uroplasma in both sample types, placenta membrane and placenta tissue. However, in the babies who were born in first 2 or 3 days of life, we did not note any uroplasma in those samples. So overall, Acinetobacter was the most commonly identified organisms in neonates between day 0 to day 28 of the age. The key finding from the study is that the higher proportion of probable nosocomial acquired Acinetobacter associated deaths in neonates greater than day 4 of the age. Klebsiella and E. coli are also considered to be the important contributor pathogens identified in the tissue of neonatal death cases. GBS was rarely found in these samples, GBS as well as uroplasma, that shows that there is no maternal transmission while the baby is in the womb. Acinetobacter was rarely found in placenta of this deceased neonates, while the uroplasma was the organism most commonly found. This study is recently published in CID, and I would like to thank colleagues at Department of Microbiology and CHS. Thank you.
Thank you so much, Najia. Wonderful study, especially considering neonatal mortality is a serious concern. So our next free paper is by Dr. Rifat Bushra. She will be talking about the pooling of urine specimens for diagnosis of chlamydia trachomatis infection by PCR in a population of low prevalence, a cost-saving measure for epidemiological and screening program. This is going to be fascinating.
Bismillahirrahmanirrahim. In the name of Allah, the most gracious, the most merciful. My name is Dr. Rifat Bushra, resident microbiology, Armed Forces Institute of Pathology. In this session, I will be presenting the research, pooling of urine specimen for the diagnosis of chlamydia trachomatis infection by PCR in a population of low prevalence, a cost-saving measure for epidemiological and screening program. I have no conflicts of interest. According to WHO 2019, the global prevalence in women was about 3.8%. It is a sexually transmitted infection. Most people with chlamydial infection are asymptomatic. However, it can cause cervicitis and urethritis in women, urethritis and proctitis in men. Its complications in females include pelvic inflammatory disease, infertility, ectopic pregnancy, and pelvic pain. It can also be transmitted perinatally from mother to fetus, resulting in ophthalmia neonatorum. According to the CDC guidelines, yearly screening in females, younger females with risk factors is recommended. However, in the pregnant females, screening in the first prenatal visit and during the 3rd trimester is recommended. The diagnosed cases are to be retested again 3 months after the treatment. So, the objective of my study is to detect a cost-saving method for the diagnosis of chlamydia trachomatis infection by PCR that is most reliable. This study will help us to find a cost-effective method for the detection of chlamydia trachomatis in low-prevalence country like Pakistan, where there is no routine testing facility available.
It was a cross-sectional study which was conducted in the Department of Microbiology, Armed Forces Institute of Pathology over a period of 1 year. The sample size was 65 and non-probability convenience sampling technique was used. This was the inclusion and the exclusion criteria. Samples were taken from the male and the female patients. The data was collected in the data collection form, and the first cache urine was collected and was transported to a department with the minimum delay. This is the biodata form. Pools of 3 urine samples were created. Pools of 5 and 10 urine samples can also be made, but they can result in false negative results. So according to the literature, pool of 3 urine samples, they give more good results. PCR of the pools were done, and any pool tested positive was then again tested individually, and data was analyzed. This is a pie chart showing the gender-wise distribution. Total sample size was 65. In the total 22 males, 4 came out to be positive and 18 were negative. However, in females, only 1 female came out with 1 intermediate result and rest were negative. We made 22 pools out of the 65 samples, and 4 came out to be positive and rest were negative. So the cost analysis. The cost per test came out to be 1,850 rupees. If individually PCR was conducted on the samples, then the cost, whole study estimated cost was about 1,20,250 rupees. However, pooling resulted in the reduction of rupees to 68,450. This resulted in cost reduction to about 43.01%. Now the literature review. A study in 2017 conducted in China where the pooling technique was used and PCR was taken as the gold standard, the cost saving was about 57%. Similarly, a study conducted in Catalonia, Spain in 2020, where again the PCR was taken as a gold standard and 3 pool sample technique was used, the cost saving was about 33%. Our study concluded that the pooling technique reduced the number of tests performed on our samples, thereby resulting in a significant reduction. We recommend the pooling technique to diagnose this infection as an effective and accurate method which is cost-effective as well as time-saving in areas of low prevalence. These are the references that I've used. I thank you all for your question.
Thank you so much. This was fascinating and such a useful piece of information that you've shared. So at this point, I would like the chair to come and conclude the session and mention the highlights that she. Thank you, Dr. Bush.
Thank you, Javeria, and thank you, organizers, for having us here. This was an excellent, excellent session, very relevant to the problems we are facing today. All the talks were very, very useful and enlightening, and all the presentations were high class, I would say, very relevant to, again, the problems we face. So do you want me to summarize the key points of presentations or just the free papers?
I think we've had a good panel discussion.
Yeah, so I will stick to the papers. So the first presenter, Dr. Rabia, actually presented a validation study. So this is a commercially available kit, but looking at pathogens which are common in our setup. So perhaps what you presented today is something which we will be using more often in the future, and this will have a place in the emergency setting when the patient comes in. And actually it will lead to a considerable cost saving for the patient, because it will decrease the days of hospitalization, the amount of empiric antibiotics used, and it will actually prognosticate on arrival. So that is something which we should look at and should explore as having a very useful diagnostic test, very well conducted study and very much needed. The second presentation by Najia, again very important because Pakistan has a very high neonatal mortality rate. And again, we know from our experience that it is usually the gram-negative rods which are the cause of mortality in most instances, but your study actually substantiates it, looking at it scientifically, that indeed it's Acinetobacter or Klebsiella, and probably poor environmental factors and poor IPC practices when these low birth weight infants are born. And if the hospital is not clean enough, sure, the baby will acquire the infection. And it's not mother to baby transmission, and we've seen it before as well that group B streptococcus has never been a major issue in our population. It's the nosocomial environmental organisms. So you've actually substantiated that, and now we have this, in a scientific manner, this piece of information. And the last presentation, again, very useful. In fact, very practical for any microbiology lab, because chlamydia, actually, it is an assumption that we have low prevalence of chlamydia and Neisseria. So once you start testing for it, then you will know the true extent. But at the moment, looking at the results, this is a very good strategy to save costs and get to the diagnosis. So all three were really good papers, so I don't know how to mark you on it, on relevance.
Tomorrow. We'll know tomorrow.
Oh, okay. So I will not announce my oh, good. That makes my life very easy. Thank you very much.
So thank you, everyone. I think we're going to close the session. There's tea and there's the exhibition for you to enjoy. Thank you.